Cek. Cek 1 2 3. Halo. Cek.
Diinformasikan kepada seluruh peserta dan juga partisipan yang masih berada di luar ruangan agar segera mengambil tempat di dalam karena sebentar lagi acara akan dimulai. Sekali lagi, bagi seluruh peserta dan juga partisipan yang masih berada di luar ruangan, kami persilahkan untuk mengisi tempat yang telah disediakan. Sebentar lagi acara akan segera dimulai.
Kami ucapkan selamat datang kepada seluruh peserta konferensi pada hari ini. Kepada Bapak dan Ibu yang baru saja masuk ke dalam ruangan, dimohon untuk bisa mengisi kursi yang kosong di depan. Sekali lagi kepada seluruh peserta yang baru saja tiba di tempat, dimohon untuk bisa mengisi bangku yang kosong terlebih dahulu di depan. Terima kasih. C,
C,
C, C, C, C, C, C, satu dua dicoba C.
C. Honorable guests, ladies and gentlemen, may we have your attention please. The ceremony is about to begin. We kindly ask you to resume your seat. Thank you.
Honorable the Minister of Agriculture of Republic of Indonesia, Honorable General Director of Indonesian Agency for Agricultural Research and Development, Honorable Director of Indonesian Cereal Research Institute,
Honorable Rector of Hasanuddin University, Honorable Rector of Dwi Jendra University, Honorable Rector of IPB University,
Honorable Keynote Speaker, Director of Genetic Resources Program CIMIC, Mexico, Honorable the invited speakers from Canada, United States of America, Taiwan, and Japan. Distinguished guests, participants, ladies and gentlemen.
Assalamualaikum warahmatullahi wabarakatuh, and good morning. First of all, we would like to say thankfulness to the Almighty God for the mercies and guidance, so we can come here to attend this moment.
With our warmest greeting, welcome to the opening ceremony of the Second International Conference on Sustainable Cereals and Crop Production System in the topic with theme, Strengthening Agricultural Resources Management to Support Food Security and Industry 4.0.
Organized by International Cereals Research Institute, Indonesian Center for Food Crops Research and Development, Indonesian Agency for Agricultural Research and Development, Indonesian Ministry of Agriculture. Harper Hotel Makassar, 23rd of September 2021.
Ladies and gentlemen, the offline participant, please stand up, and the online participant, please keep sitting. Singing the national anthem, Indonesia Raya. Sit down, please.
Akbulo Sibatang. The diversity of culture reflects the reach of ethnics of South Sulawesi Island, which is settled by many kinds of ethnics. There are 4 ethnics which are interacting to each other, namely Buginis, Makassaris, Mandaris, and Torajanis. They are tightened together and make them stronger even though they are in different languages and traditional customs. They also live in peace together with Tionghoa, Arabian, and Indian ethnics as an implementation of unity which is reflected in an art creativity called Akbulo Sibatang.
Ladies and gentlemen, we'd like to present Akbulo Sibatang dance presented by Patondro Art.
La
Malakolay malanirikato
Honorable ladies and gentlemen, our next agenda is reporting speech by the chairman.
Dr. Muhammad Azrai as the Director of Indonesian Cereal Research Institute.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Honorable Director General of Indonesian Agency for Agriculture Research and Development, Rector of Dwijendra University, the deans of the Faculty of Agriculture from several universities, distinguished invited speaker from Canada, USA, Taiwan, Mexico, and Japan. Ladies and gentlemen, first of all, I would like to thank God for His blessing so we can meet offline and virtually at this conference. I would like to welcome all participants who attend this conference. This is the second IGFST, and we have successfully held the first IGFST in 2019. This event aims to be an exchange information platform of research of corn and other food crops, as well as the challenge faced in development. This international conference is also an opportunity to establish cooperation in the development of crops with several research institutions, bilateral and multilateral. The International Conference on Sustainable Cereals on Crops Production System in the Tropics is organized by Ministry of Agriculture and the Indonesian Agency for Agricultural Research and Development, supported by PAIUI, SIMMIT, Dwijendra University, Breeder Organization Profesi, and Agronomics Organization Profesi. The theme of this conference is strengthening agricultural research management to support food security and industry 4.0. This international conference is held on 23rd and 24th September 2021 at this Hotel Harbour, Makassar. This conference presenting the Director General of IRD and Director of SIMMIT as keynote speakers, as well as invited speakers from various fields such as Bunyamin Taran Professor from University Saskatchewan, Dr. Kerti S. Rattle from Texas E&M University, Dr. Juan Landiver Bowles from Texas E&M AgriLab Research, Yu Xinning from National Chongqing University, Taiwan, Dr. Naori Miyazawa from Nagoyama University, Japan. This conference attended by limited of 100 offline participants, such as lecturers, researchers, agriculture extension, private company, and millennial farmers. This event is also held virtually via Zoom meeting with over 1,000 participants from Indonesia and abroad. This conference is arranged in plenary and parallel session. A total of 115 papers from various disciplines, including plant breeding, crop production, system, pest and disease, post-harvest, seed production, and social economy will be presented. Finally, we would like to thank to the keynote speakers, invited speakers, oral speakers, and other participants for the contribution. We also give thanks to ICRI researchers for the research royalties as the main sponsor of this to the second ICFST. Once again, I would like to congratulate the committee for organizing this international conference and hope we always in a very good health. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Distinguished guests, ladies and gentlemen, the next agenda is recording opening speech.
The General Director of Indonesian Agency for Agricultural Research and Development, Dr. Fajri Jufri.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning everyone, distinguished guests, invited speaker, ladies and gentlemen. I would like to begin by giving thanks to Allah Subhanahu wa Ta'ala. His blessings are evident as we gather here today to take part in the event, the second international conference on sustainable cereal and crops production system in the tropics. My warmest welcome to all you this conference. It is our honor to present at this conference event. I hope this event will bring such contribution toward the development of agriculture, particularly in Indonesia. Furthermore, this event is supposed to be a media to exchange information among participants from different countries regarding technology development, input crops, and other agricultural sector. Ladies and gentlemen, the challenge to sustainability has nourished Indonesia in the world population. Meanwhile, the impressive progress that Indonesia and the world have achieved in reducing poverty since few decades ago, temporarily set back by the COVID-19 pandemic, also means that per capita demand for agricultural production will outpace population growth for decades to come. This is called for rethinking agricultural strategies, because continuing or increasing the same effort of recent decades will lack yield diminishing return. various novel approach, tool and knowledge offer option for designing agriculture for a sustainable future. Distinguished ladies and gentlemen, as the effort to compromise this challenge, the Minister of Agriculture, Republic of Indonesia, has compiled a wide range of policies and programs, among others, increasing the productivity of staple food, particularly rice, facilitation food distribution, facilitating access to transportation, maintaining the food price stability, developing the buffer stock and market intervention. Ladies and gentlemen, we believe that key tools to help to take advantage of the agriculture potential are related to knowledge and innovation transfer, consulting service, cooperation of research and development, as well as science with particular emphasis on the significance of smart farming application in order to support Industrial Revolution 4.0. Strategies on implementing 4.0 Industrial Revolution can be done by strengthening collaboration between the Ministry of Agriculture and the university or college as well as the industry. Ladies and gentlemen, it's my hope this conference will address itself to this issue critically and come up with some recommendation which will help to accelerate agriculture innovation and implementation in Indonesia. Finally, I wish you a fruitful and pleasant international conference and pray that this event will become a pioneer for continuous and long-lasting mutual bonding and networking in the year to come. Thank you very much. And Bismillahirrahmanirrahim, I declare the conference operational open. Assalamualaikum warahmatullahi wabarakatuh.
Distinguished guests, ladies and gentlemen, we are honored to have a keynote speaker,
Dr. Kevin Fixley, Director of Genetic Resources Program, CIMIC, Mexico, and the chairman of this session is Dr. Willy Bayuardi Suwarno, SPMSE from IPB University.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A very good morning, everyone, distinguished guests, ladies and gentlemen. It's an honor for me to be with all of you as a moderator in this keynote session. And this very important session, we will learn about the global agricultural resource, which is a very important topic for us. Let me introduce our keynote speaker, Dr. Kevin Fixley. Kevin Fixley is a graduate from Purdue University, the University of Florida, and Iowa State University. He got his PhD in plant breeding, and his current position is a Deputy Director General for Research, Breeding, and Genetics, and also Director for Genetic Resources Program at the International Maize and Wheat Improvement Center, CIMIT. He also lectures on agriculture and nutrition and health each fall semester at the University of Wisconsin-Madison, USA. He is also a part of the leadership team guiding the design of the research portfolio on genetic innovation for CGIAR. And Kevin has a research interest, including equity in access to best that science can offer. So without further ado, ladies and gentlemen, please join me in welcoming Kevin Fixley.
Thank you very much, Dr. Suwarno. It's a tremendous honor to be invited to speak. Just to notify, I need to be given permission to share my screen so that I can do that. And while that's enabled, I would like to thank the DG of IARD, also the organizing committee, and my friend and colleague, Dr. Azrai, for the invitation to participate in this international workshop congress. I'm still unable to share my screen, so perhaps you'll need to project from your side. Okay, now I have suddenly been given permission. Thank you. Let me go ahead and do that. Go into presentation mode. So I hope you can all see my screen now. If not, please let me know. So as I was saying, it's a great privilege to open this Congress with my keynote presentation. I'm also very glad to have as co-author Dr. Barbara Wells. She is the director of the Genetic Innovation Science Group of CGIAR. And I'm going to take this opportunity to share with you what we are doing in the CGIAR as we redesign the agricultural research that we do with you and with other partners around the world. I will probably share about half of my presentation will be explaining what we're doing, and the other half will be giving examples of the science that we're doing. So I hope this will be of interest to all of you who want some science, but also who want to see the opportunities for collaboration with the new group of CGIAR. For those of you who aren't familiar with CGIAR, it's the group of international research centers, CIMMYT, CIAT, IITA, SIP, IRI, International Rice Research Institute, and others who are now basically merging. We're coming together. And the reason we're coming together is to be more efficient and more effective, addressing the challenges and working with our partners, such as yourselves, to address the challenges that we confront as society. Well, let's see, I'm not able to advance my slide. There we go. So the first challenge, and I won't spend much time talking about these, but the first challenge is the growing population. We expect to go from today's 7.5 billion to at least 10 billion by the end of this century. So we're going to have many more people to feed. We also have a remaining problem of poverty. And this map shows you, it's an interesting way of looking at the world, where you see relative to size, you see the size of countries on the map. And what I'd like to point out is that if you look at the bottom part, under 5 mortality and underweight prevalence, which are indicators of malnutrition, in these maps, Africa and South Asia look huge. But above, in the map that indicates relative wealth, you see that Africa is hardly visible and South Asia is very small. So clearly, malnutrition, undernutrition is associated with poverty. And one of our main objectives has to be also reducing the prevalence of poverty. We have had quite a bit of progress. These graphs show that in all regions of the world, there has been tremendous progress over recent years in reducing poverty. And with reduction in poverty, there have also been reductions in hunger. So you see that the number of people hungry or undernourished has also declined over recent decades. And as mentioned by the DG of IARD, we're now facing a setback due to COVID, which is causing tremendous pain and difficulties which will last for many years to come. So the challenge from poverty and malnutrition will remain with us, unfortunately, for many years. The third challenge is, of course, climate change. And I don't need to tell this audience about the rising temperatures. You can see on this map the expectation of the difference between the end of this century with the end of the previous century in number of degrees centigrade expected increased temperature. And most parts of the world are expecting somewhere in the range of 2 to 3 or 3.5 degrees centigrade, according to this model. So it's definitely getting hotter. And of course, for agriculture, this is mostly bad news, particularly in the tropics, where many crops such as rice, maize, and soybean are in fact susceptible to increased temperature. And we expect, if we do nothing through breeding and agronomy and other mechanisms, we will experience very large reductions in yield. So this is an important constraint. Also, nutritional content often declines along with increased temperatures. So climate change is the third important challenge. And finally, the one which the CGIAR is focusing increased attention upon is that we have to address these challenges in a sustainable way. We need to ensure that agriculture does not have a time limit, that we do not use the resources of the planet, and that we can continue to produce and sustainably produce food and nutrition for the population of the world going forward. And this landmark paper from Nature in 2009 highlights that in terms of biodiversity, in terms of nitrogen cycle, in terms of phosphorus cycle, we're already beyond sustainability. And so we need to be finding ways to reduce and to improve these areas, which many of them are impacted by agriculture. Now, in the past, we've had some success. And the first one I want to present is from Norman Borlaug, who indicated what we now call the Borlaug hypothesis. And in this graph, what you can see is the amount of land, so number of hectares, that were utilized in 1950 to produce the cereal production of 1950, which was 600 million hectares of land to produce about 650 million tons. In the year 2000, we produced about almost 2,000 million tons of cereal grains, and we used only slightly more land, 660 million hectares. So by improved technologies, by improved genetics and agronomy, we managed to save 1.1 billion hectares of land. And this is a tremendous contribution from agriculture, and we will need to continue to replicate this success going into the future. One of my colleagues, Frederic Baudron, illustrated for other countries, India, Mexico, and Kenya, similarly, how much land has been saved by improving technologies. So it's similar to the previous graph. You can see that in Mexico, tremendous amount of land savings because of improved technologies producing maize, also for Mexico, wheat, for India, wheat and maize, and also considerable savings in the case of Kenya. So we're now redesigning the CGIAR agricultural research portfolio. And I would say the biggest change that we are making is that we are placing increased emphasis on this component that I'm showing you at the bottom left, which is the agronomy, the resilience of agri-food systems, the cropping systems that must be sustainable for the long term. So I'm going to explain briefly what we're doing in each of these. I expect that each of these will be of interest to some of you in the audience. And then I'm going to spend most of my time speaking about genetic innovations, the genetics, the breeding, which is where I have been personally involved. But each of these efforts, both in system transformation, in genetics, and in cropping systems, are going to have impacts in these 5 areas: nutrition and health, poverty reduction, gender equality, climate adaptation, and environmental health. So primarily those problems, those challenges that I mentioned in my first slides. The final component, which I'm not going to talk too much about, are regionally integrated programs that will bring together the contributions that technology offer from each of these 3 science groups into packages that are tailored for each of the regions in the world. So let me tell you first a little bit about systems transformation. This one is primarily about policy, about issues in policy. And recall that we often say that the progress in yield, the progress in productivity, is attributable in equal proportions or approximately equally to agronomy, genetics, and policy. So this policy component is incredibly important. And I've put a star next to those topics which are going to be addressed by this systems transformation science group, which overlap in mandate with genetic innovations or with the breeding components. And you'll see that we're going to be working together with them on resilience against climate change, on reduction of greenhouse gases by producing crops that serve as better sinks of greenhouse gases, We're going to be thinking about market chains and how we can make crops, varieties that are also more profitable for farmers to grow and more demanded by the market. Healthy diets will be, of course, important, use of digital technologies, and a lot about policy for strategies to improve the effectiveness and the sustainability of cropping systems. So that's for all of you who are socioeconomists and would like to work with CG on systems transformation. The second group is what I would call the cropping systems or resilient agri-food systems. And this has three main themes and areas, and many areas in which we, the genetic innovations or the plant breeding group, will work together. For example, on plant health. We know that with climate change, there's going to be changing spectrum of diseases and pests and increasing pressure that we will need to address through breeding, genetics, as well as through agronomy. We're going to be developing better varieties to feed animals, so forage production that will enable sustainable animal production. Probably some work on biofortification and nutritional enhancement to improve the One Health component. Mixed farming systems. We need to have more diversity in our farming systems. And of course, climate resilience is a common theme throughout the 1CG objectives. So the third science group, and I'm going to talk to you and give some examples and some science from each of these areas in the coming slides, is about genetic innovation, or essentially the plant breeding. And it's divided into these 6 thematic areas that you see here. And again, I will explain each, but first overview is that market intelligence is where we're going to actually define the targets, define the priorities. And we need to work with you to define what are, in fact, the varieties that are needed and in demand for rice, for maize, for cassava, for bean, for all the crops of Indonesia. Of course, we will maintain the gene banks because the conservation of genetic diversity in perpetuity is critical. We're going to have several themes around plant breeding, the actual breeding group, the use of enabling technologies that facilitate more effective breeding. And I'll tell you a little bit about the work that we're doing in genome editing and transgenics, also within precision genetic technologies. And finally, the last group is about reaching farmers with improved seed. We have a lot of work to do on many of our crops in more effectively reaching farmers that still are not benefiting from the best that science can offer, that still are not able to use or to access improved varieties. So let me begin talking a little bit about market intelligence. The idea of this group is to work with extensive groups of stakeholders. It will include the food industry, it will include the ecologists, it will include, of course, many farmers, it will include seed companies, to really define what is the demand, what are the traits that are needed in the market so that we can have the greatest impact on these 5 areas that I mentioned to you earlier. So quite a bit of market research to better focus the investment that we do in plant breeding. And with this intelligence, we will then define what we call product profiles. And the product profile is just a description of the varieties that are needed and that must be developed by our breeding teams. And those will guide the efforts of our breeding teams. This intelligence will also allow us to develop investment cases that will be attractive for funders to be able to say, yes, we want to invest in cassava for West Africa because it can have tremendous impact. Or we want to invest in rice for Indonesia because that's how we can achieve the greatest impact. And finally, they will also do our impact assessments to make sure that we are in fact being effective. So some ways that some data that they will use to prioritize, you can see on this graph, for example, production area and value of production. So we know that in South Asia, rice, wheat, and maize, in that order, are the crops that are grown most extensively. And in terms of crop value, it's also rice, wheat, and maize. So of course, the area and the value of production will be important determinants of what investment we make in such crops. Nutritional importance. How much of the protein or how much of the energy do each of these crops provide for populations? And we've done similar analyses for micronutrients. How much of the iron, zinc, or provitamin A do different crops supply? And for example, in sub-Saharan Africa, we know that maize, wheat, and rice are important. In South Asia, rice, wheat, and maize, but also bananas, potatoes, and cassava are tremendously important crops, as are soya, beans, and peas in some areas of South Asia. We will look at stakeholder preferences, we will look at market opportunities, sustainability of cropping systems, and all of this information will be used to help prioritize the product profiles and the breeding objectives. Also look at where we can have the greatest impact in terms of numbers of undernourished people. And we can see that probably South Asia and much of sub-Saharan Africa are areas where there are large numbers of undernourished, and it coincides with where we have large numbers of poor who cannot afford to purchase a more balanced, more nutritious diet. So these areas will, of course, and you see Indonesia is also, unfortunately, sharing a fairly important proportion of undernourished. So we would also be interested to work in Indonesia. So the second area, remember there are 6 of them, is gene banks. And this one is fairly intuitive. Of course, we need to guarantee in perpetuity the availability of genetic diversity. This is, in fact, the basic building blocks for all plant breeding. It's the genetic diversity that breeders need to address the problems, the challenges of today and of the future. So there'll be tremendous investment in maintaining the diversity, but also in supporting breeding programs to increase the value and to use those collections. So I want to give you a couple examples of what we're doing and what we could do, for example, with you in Indonesia. This first example is a project that we conducted with a bank in a city called Guadalajara in Mexico. It's a germplasm bank that also collects maize. And this is the particular maize landrace that we were interested in this study. And they were interested to know whether their collection was duplicating CIMMYT's collection or it was complementing our collection. So we used genomic data, and we were able to construct this analysis. And you see everything in yellow are accessions in the gene bank of Guadalajara, and everything in blue are materials that we have in our collection at CIMMYT. And quite to our pleasant surprise, we discovered that these are not duplicating, that in fact almost all of the collection in Guadalajara is different from what we have at CIMMYT. And so we were able to conclude that they are investing resources wisely in maintaining this collection. Another project was with a small seed company called Novasem. They were interested in knowing how to utilize CIMMYT germplasm in their breeding program. So again, we used genomic data, and we were able to do an analysis to look at genetic distances. And you can see in red some of the elite maize lines from CIMMYT, and in the green are the elite lines from the company Novasem, and in yellow are some publicly available inbred lines from the United States. And based on genetic diversity, they were able to predict which lines to group in terms of heterotic groupings in order to maximize the effectiveness of their breeding program. And this is something we would be able to do together to look at your materials and help design breeding programs to make use of CIMMYT or other germplasm within your programs. Then if we move to the third initiative, which is the actual breeding, where the breeding work is going to happen, they're spending a lot of effort. I would like to highlight this third one, transforming the relationship between CG, NARS, and small and medium enterprises or small seed companies. And the vision is to have truly collaborative, truly shared breeding programs where the responsibilities for different parts of the breeding work are shared among the different partners in these networks so that we can also have efficiencies and also avail some of the most modern tools that I'm going to tell you about in the coming slides. There's also a very important section on trait discovery, which is how to utilize the materials in the germplasm bank, and I'd like to tell you a couple examples about that work as well. So I think most of you who are plant breeders are familiar with genome-wide association studies, or GWAS. This is nothing new. It involves use of detailed genomic data, usually tens of thousands or hundreds of thousands of markers on the genome of every line or variety in the trial. It involves extensive phenotypic trials, which are incredibly expensive. When you have to grow trials of large numbers of accessions at multiple locations, it is very, very expensive, and in fact, prohibitively expensive. And when we combine these data, we can do an analysis. You see the traditional Manhattan plots. You can do associations of those genomic, we call them SNPs usually, the single nucleotide polymorphisms, with different phenotypes, and you can identify SNPs or quantitative trait loci that are highly associated with the type of trait that we're interested in, which might be drought tolerance or high zinc or any other trait, and that gives us an indication of which areas in the genome are important for use in breeding programs. As I mentioned, this is old technology and it has been useful, but we now have a new idea, and this is called environmental GWAS, or environmental genome-wide association studies. This is, I'll give you an example on the next slide, but the concept is illustrated on this cartoon. We take the same genomic data that we have for about 17,000 maize accessions in our germplasm bank, and then using the passport data, we know exactly where each one was collected. We can look at historical environmental data for those locations, data about soils and data about climate, and we can look at, for example, which of these land races were collected from areas with high temperatures or with low zinc in the soil, or in the case of the following example, with lots of salinity, with high salinity problems. And you can do an association study and identify which QTL or which SNPs are associated with the varieties that, or the land races rather, that come from those regions. And this gives you an indication of which genomic regions are important for success in these stressed environments. And so here's an example, quite an exciting case. We were interested in finding germplasm in the bank that could be useful for salinity tolerance. So we identified 524 land races that were collected in areas with severe soil salinity. We then used genomic data to select 281 that avoid duplications in terms of genomics. So a very diverse group of 281 that were representative of those 524, which were a selection from more than 17,000. We then took those 281 to the glass house, where we evaluated them in 3 levels of salinity, and we were able to identify which ones appear to be most tolerant to saline conditions, and those were then taken for validation in the field. So this saves tremendous amount of time and resources. A great exercise, and we could do this also with you. In terms of also accelerating breeding, we're working intensively to shorten the breeding cycle, the time that it takes to go from a breeding cross to selecting the best lines to make the next generation of crosses. And using some of these technologies, in the case of wheat at CIMMYT, we're using rapid generation bulk advances. We've managed to cut our breeding cycle from 6 years to 3 years. And of course, this would result in expected tremendous gains over time. So the genetic gain in years or per year will be nearly double of what it would have been before. Something else we're doing, and these 3 pictures at the bottom of the graph illustrate our speed breeding facilities. This is specific facility where this picture in the middle with the purple colors, that's not a bad picture. That's actually the light, special light spectra that help the plants to grow more rapidly. They're given very long duration days, and the result of these methods and technologies is that we can produce in 90 days seed to seed for crosses, and in 65 days seed to seed if we're just advancing generations. And this will allow us to get 4 generations per year instead of 2, which is the normal cycle. So these are exciting new technologies that we're using and that we'd love to share with you. This slide is primarily about how we intend to work increasingly as partnerships, collaborative breeding programs with national programs and small and medium enterprise companies. The next one I'd like to share with you is the 4th initiative, and this one is about enabling technologies and services. And this is all about helping us to modernize our breeding programs. A lot of gained efficiencies by sharing services. For example, why should each of us have a molecular laboratory when we can have one consolidated laboratory that does all the genotyping that's essential for many of our procedures, so we can have tremendous savings by doing so. So if we look at some of the examples, here's a case where we use genomic data to predict which crosses to make in a wheat breeding program. So you can see that there were 70,000 possible crosses among 376 lines. And of course, nobody can do 70,000 crosses. So we did a small subset. We just did a few hybrids, evaluated those, and using genomic data, we're able to predict what would be the likely yield of those other crosses that were not done. And we actually found a very high R-squared, a very high correlation, very possible to select those which would be most likely to be successful and then only make those crosses that are predicted to be successful. A lot of work with new experimental designs, especially looking at partial replicated designs, looking at augmented designs, looking all sorts of experimental designs and biometrics that will allow us to be more efficient. And the most important application of this is the following slide, which I'd like to show you. We are going to now, we already have actually increased tremendously the number of on-farm trials with farmers to evaluate qualitatively what are the traits that they appreciate, what are the varieties that they would most like to see made available to them. And this was made possible by switching to large plots with very few plots in each farmer's field. In this example, there's only 6 plots that a farmer is asked to grow. It's an incomplete replication, but by having many, many locations, we achieve adequate replication. And of course, for this, we have to use very good experimental designs. The last example, it just illustrates the complexity of the big data that we are collecting today. We're using drones, we're using genomic data, and we're using extensive phenotypic data, and it's a superb challenge to bring all these data together and to actually facilitate decision-making in breeding programs. Most breeders are no longer capable of doing this. I would be unable to do this using Excel and using SAS. My own computer would not be able to handle these data, nor would I know how. So we're developing software tools. We're calling it Enterprise Breeding System, also the Breeding Management System. So these are software tools that help us to collect, to curate, and to co-analyze these data for timely decision-making for our breeding programs. I'd like to spend a few minutes talking about precision genetic technologies, which is the 5th of our 6 thematic areas in the genetic innovations group. And it's primarily about gene editing and transgenics. But I would say the future really is gene editing and probably much less work on transgenics going forward. So just a few words about what we're doing in gene editing. Of course, I think we're all familiar, hopefully you're all familiar with examples from the literature of what has already been validated and demonstrated in terms of traits. And here you can see disease resistance, enhanced nutritional quality, drought tolerance, salinity tolerance, control of parasitic weeds such as this one that the gentleman is holding. And nutritional enhancement. All of these are traits that we can address through gene editing. And this slide illustrates projects that we are actually doing in the CGIAR, in the different centers that are now coming together into one CG. We're working on banana diseases. We're working on cassava diseases and quality. We're working on rice diseases and also safety, food safety issues, and nitrogen use efficiency. Working on diseases of maize and wheat, as well as diseases of potato. So many opportunities we're already pursuing in these gene editing projects. And here's just one example from maize, where you can see the effect of one allelic change in maize. Here you have, of course, the resistant, which has the favorable allele, and the susceptible with an unfavorable allele. This has been traced through work by several colleagues at Corteva and CIMMYT, and we've identified the gene which is causing this. And we've already designed edits that are effective in producing this effect in elite lines. So we are now editing elite maize lines to confer resistance to maize lethal necrosis without affecting any other traits in these elite materials. So tremendous opportunities with gene editing. But I spend a lot of my time thinking, philosophically about what we can do and who will benefit from these technologies. We need to notice that gene editing is, in fact, in many ways different from transgenic. There are differences in terms of who's working with these traits. And there's also differences with what crops are being worked upon. And because gene editing is relatively affordable, and it's something that can be done by many laboratories around the world, it is something that is being used for crops that generally are not targets for transgenic research. Transgenic research is very expensive, and so it can only be done for crops that are going to provide a where gene editing is already being used to develop useful traits. And on the left-hand side, I find it very interesting because one of the common criticisms of transgenics is that only large companies are able to invest in transgenics. But you see that for gene editing, most of the research is being done by small private companies. And quite a large amount is being done by public sector and universities. And the component for the large private is only about 10%. And also something that's different is that the owners of the technology, the Broad Institute and Corteva Agri-Science, have made an agreement, which you can read about on this link below, to make this technology available for developing uses for smallholder farmers in low and middle income countries. So gene editing is a potentially valuable technology because of the reasons I've explained. It's relatively accessible. It's something that we are doing in the CGIAR because we believe that the technology should be available for those who would like to use it. It can address many of the important goals and challenges that I mentioned in my introductory slides. The last of the 6 initiatives that I'd like to just mention briefly is about seed systems. All the wonderful work that we do means nothing if farmers don't benefit from it. So there's a lot of models that need to be designed and validated so that we can actually have and reach more farmers with improved varieties. So this is perhaps in many ways the most important of our initiatives, because it is the initiative that we depend upon to reach farmers and to achieve the success that we want to achieve. So to summarize my final slide with the key points, I've pointed out, I think we're all aware that the challenges are greater than ever in terms of feeding the world and doing it sustainably. So the science of the past, it isn't going to be good enough. And this is why the CGIAR is merging. We are reorganizing. We're putting much greater emphasis than before on sustainability of cropping systems and using the best science that's available and making it available to all of our breeding programs, and also putting a lot of effort into prioritizing together with stakeholders and partners to make sure that we invest in the most potentially impactful gene editing, but variety development efforts. Finally, and most important perhaps for this group, for this Congress, is that we are in fact exploring improved models of collaboration, and we very much look forward to talking with you about how we can work together to share and to learn together and to do a better job of addressing these tremendous challenges. With that, Mr. Chairman, Dr. Suwarno, I'd like to thank all participants. And of course, you see our contact addresses. We'd love to hear from you. Over to you. Thank you very much.
Thanks very much, Dr. Bisley. So, thanks very much for the enlightening presentation for all of us, Dr. Pixley. We received 2 questions from the chat box, and let me read the question, and perhaps Dr. Pixley can please address the question. The first question is, the adoption of novel technology for improving crop production, for example, molecular breeding program and utilization of genomics information, is still unaffordable for developing countries like Indonesia. What kind of strategies can be implemented to reduce costs and promote up-to-date breeding tools? And how CGIAR play a role? Yes, Kevin.
All right, thanks. Yes. Thanks for that question. In fact, this is what our, I forget what number it is, number 4, I think, thematic area is about. And that is about sharing these tools. It is expensive. And this is why if we come together, first of all, the CG breeding programs, we have great bargaining power, because now instead of CIMMYT going to a genotyping company and saying we would like to sequence 10,000 genotypes per year, now the CG comes together and says we would like to sequence 1 million genotypes per year. And you get a much smaller price, so a much more affordable price for these services when you come together as a team. And the second part is if we work as a collaborative network, then we share the expenses. We might be able to contribute to genotyping costs for you, and you would then contribute to some of the field evaluation costs for the materials for the breeding program. And we, of course, share the data from both with each other. So I think there's tremendous opportunities for synergies and for sharing of the expense, as well as reducing the cost by working in larger groups together.
Thank you. So another question is about the ideas about one CGIAR. Let me read the question from Bu Nuning. I'm particularly interested in this one CGIAR idea. For your information, currently the government of Indonesia intends to unite all of its research institutions into one. I want to know the opinion from you, Dr. Pixley, from the research institute's unification efforts. And are there any particular recommendation based on your experience in supporting one CGIAR? Thank you.
Thanks for that question. We're in the middle of the process now, and officially the starting date is going to be 1st of January of next year. And I can assure you that there's a lot of stress, there's a lot of hard work, and there's a lot of uncertainty right now that we're all dealing with. This is not easy. You can imagine when two companies merge, it's complicated, but when 12 research centers merge, it's very complicated. And my experience or my advice is, at some point we just have to decide that we're going to do it. And it's not going to be easy, but we are going to do it. So we have to stop, we had to stop thinking about, well, should we do this, should we not do this? And just basically draw a line and say, yep, this is going to happen, so now let's make it work. And we accept that it's going to be very challenging. Next year is going to be a difficult year, and we have to have some opportunity to duplicate. And I think in the first months, the first year, it's not going to be so efficient, because we probably will have some duplicated services, some double cost, for example, for human resources services. But that will be worked out during the first year, and we expect that by second year and third year, the tremendous advantages will show up. So I think the first step is just to decide that this is going to happen, and that we can all envision that it will be beneficial. But those benefits aren't going to happen immediately. It's going to be difficult, it's going to be challenging, and you just have to keep the end in sight and keep thinking about the benefits that can be achieved, and then work to achieve those benefits.
Thank you. Another question from Dr. Indrastuti. Is the heterosis pattern in rice similar to the heterosis mechanism in maize? And could we use the same method that you explained in the maize for hybrid rice development, meaning exploiting the heterosis? Please.
Yeah, that's a really good question, and I don't have a really good answer. Sorry to say, because for one thing, we don't really understand heterosis in maize either. I don't think anybody really can explain what heterosis. There are several hypotheses that explain heterosis. We know a lot of things about heterosis. We know that you need genetic diversity between parents to get heterosis, but we also know that diversity is not enough. It's not only genetic diversity to get good heterosis. So the experience, from maize that I think will also be useful for rice and probably any other crop is that probably the most useful thing that was done in maize, particularly in the United States, was at the beginning to define these are going to be our two heterotic groups, and to keep those heterotic groups closed. Don't allow them to intermix, and continue to improve them and build that heterosis over years. One of the mistakes that we've made at CIMMYT is to not sufficiently close those populations. We allowed too many lines that were kind of fitting nicely into both groups, and keep working with those. And I think we would have made more progress if we had been very strict and said this is one group and this is the other, and insist on improving that heterosis over years. So that's about the best advice that I could give you, and hopefully you'll hear from some other speakers during this conference who will give you some more suggestions about how to build heterotic groups in a new crop. We're also interested in doing this for wheat. And again, it's not simple, and it will take time.
Thanks very much. One more question, perhaps. What do you think about the importance about the specialty corn in the future? This is a question from Dr. Yasin.
Yeah, no, I think specialty corn is tremendously important as an income earning opportunity. I would love to see it being used by smallholder farmers to be able to greatly improve the viability of their farms. So that, in fact, a smallholder farmer can make a reasonable livelihood by growing some of these crops. So I think that, probably it's not going to feed the world. I mean, sweet corn and popcorn and waxy corn are specialty foods that aren't staples, that we don't eat huge quantities, but they are an important source of income. And I think we all enjoy them as well, so that's why they have value. So I think there is value in investing some amount of resources into these and making them into options for for farmers to be able to have better livelihoods.
Okay. Very much, Kevin. From our session this morning, we learned that it's very important to have a strong collaboration among institutions, including international institutions, to implement our shared ideas and also to achieve our shared goals for sustainable agriculture. Let me conclude this session. Please give a big round of applause for our special speaker. Thanks very much, everyone. Assalamualaikum warahmatullahi wabarakatuh.
Thank you.
Thanks very much, Kevin.
Distinguished speakers, participants, ladies and gentlemen. For the first session of invited speaker, we are honored to have the four speakers, Dr. Bunyamin Taraan, Professor of University of Saskatchewan, Canada.
Presenting his paper, Agri-Food Innovation Chair in Chickpea Breeding and Genetics. And the chairman of this session is Professor Dr. Insinyur Suyamto Harjo Suirjo, MS from ER.
Thank you. Good morning, ladies and gentlemen. Welcome again to the second ICFST. It's a little bit difficult to remember. As we know that the first ICFST was conducted 2 years ago, also in Makassar. So it is the second international conference. Well, let me introduce myself. My name is Suyamto. I am research professor emeritus from Indonesian Agency for Agricultural Research and Development, and now as a lecturer in Faculty of Agriculture, University of Islam, Malang. So please, if you go to Malang, please come to my university. Well, I will be the chairman of this session, and we have one invited speaker from Canada. Before I continue, I would like to say hello to Professor Benjamin. Hello, Professor Benjamin. Hello. Halo, good morning Indonesian time. Pak Bunyamin, do you hear me?
Yes.
Okay, all right. Well, I will read your CV first. Dr. Bunyamin Taraan is the Ministry of Agriculture Strategic Research Program Chair in Chickpea and Flag Breeding and Genetics. He is a professor at the Crop Development Center, Department of Plant Sciences, College of Agriculture and Bioresources, University of Saskatchewan, Canada. Benjamin did his undergraduate program in agronomy from Sriwijaya University in Palembang. So actually, he is originally from Indonesia. And his MSc and PhD from the University of Guelph, Ontario, Canada, specializing in plant breeding and genetics. His main research focuses on development of high-yielding chickpea and flag varieties with superior agronomic characteristics, resistant to diseases, acceptable quality, and well adapted to Canadian environments. Benjamin has released 16 chickpea cultivars. He has published 112 articles, including 3 articles in Nature with Scopus Age Index of 27 and Google Scholar Age Index of 33. Benjamin also teaches an undergraduate class and supervises several graduate students, MSc and PhD, postdoctoral fellow, and research staff. Well, I would like to invite Pak Bunyamin, so I will call you Pak, that is more familiar. He will present about the plant breeding in the 21st century, challenges and opportunities. Pak Bunyamin, please, your time.
Thank you very much, Prof. Suyamto. I wish I'm in Makassar, and then we can meet again and then talk different things about research and agriculture in Indonesia. Assalamualaikum warahmatullahi wabarakatuh. So I'd like to thank you for the local organizing committee, for the Ministry of Agriculture in Indonesia for inviting me for the second time to share my view and experience working as a plant breeder in the cold countries in Canada. So I'd like to share my screen. Please allow me to share my screen to the committee, please. Okay, so thank you again. So what I'll be talking this morning in Indonesia, which is 8 p.m. in my place in Saskatoon in Canada. So I'll be talking about the plant breeding in the 20th century. This is about the kind of a review, the opportunities, where we are now, what the challenges are, and then a few examples here and there, what I'm doing here at the University of Saskatchewan. So my main crop as a breeder at university, mostly on chickpeas, and recently I'm also assigned to take over to lead the crop flax, also known as a linseed. This is an oilseed crop, healthy oilseeds that Canada is producing, one of the leading producer of flax in the world. So here is my location, as you see here, far north, and I'm behind 14 hours from Makassar, as I said. So Makassar now about 10 o'clock, and I'm here 8 p.m. And this is the university where I'm working. It's very integrated in biology. Agriculture is one of the strengths, one of the strong program at the University of Saskatchewan. The province is itself mainly agriculture-based. One of the largest agriculture sector in Canada is located in Saskatchewan. So let's go back in history, back to where we are now and then where we came from. So human being, Homo sapiens, originated about 400 to 250,000 years ago. And the major crops were kind of domesticated about 5,000, 10,000 years ago. So what is interesting to see historically is that the development of human civilizations is very much correlated with the development of agriculture itself. So it's been historically known, for example, in central production where domestication is happening, and also parallel with the agriculture and the development in some areas. For example, rice and soybean in China, bean and rice and others in India, wheat, barley, peas in the fertile crescent in southeastern Turkey, and a number of other like corn in Mesoamerica, squash, bean, and potato. So happen to be now kind of located also in close to that area is the most productive agricultural areas in the modern world. So it took a lot of time for the domestication selection to increase. For example, the crop that Dr. Pixley been working, or Dr. Swarna, Vayuadi Swarna working, is corn. Like it took from 7,000 to 500 years ago from the evidence, the archaeological evidence, changing from the small to what we see corn that we eat now. So at the same time, the population of the world like now increasing. So in the last 100 years, especially, the population increased like almost like a quadruple in the last 100 years. So this is really putting kind of a lot of pressure for us working in agriculture. How we'll feed off this population of the world. Okay, so if you look at this graph here, okay, the increase of human population by 60 to 80% from now on will require about double the global food production by 2050. Okay, so we only have 30 years from now, maybe less than 30, 29 years from now to make it that double compared to in the past, like 12,000 years where people domesticating to feed the world populations. So we have not very much kind of time left. We need to do something to kind of to maintain this pace of progress. So at the same time, as Dr. Pixley already mentioned a few times, we also challenged by a number of factors environmentally. This is the data from 2018. For example, climate change. There will be an increase from 2016, about 14.7 to 16 degrees, an increase in pest amount and global production loss due to pests. There's a battle of war for water, okay, more scarce water, more limited, and also growing populations, as I just mentioned, from over 7 billion to 9.8 billion, and also less arable land at the same time. So this is the challenge that we'll put together for us in agriculture, how we kind of cope with all these challenges that we see. Okay, so plant breeders, in response to these challenges, kind of been working or develop improved varieties that kind of more or less can be divided into these three groups. The first one is to stabilize, increase yield despite the changing climate. Develop the varieties that can withstand pest and disease with fewer crop inputs, and maximize the resource utilization of those for like land nutrients and water of those new varieties. So what happened now? Okay, we have the challenge, we have kind of the goal, but for many of us knows that the plant breeding is a long process. It needs the genetic variation and selections, and then the breeder's toolkits confined to the sexual recombination between variations of the same species. And we also kind of use extended the boundary related species, the wild species, but some limited success and some unsuccessful. And very rarely interspecies sexual recombinations. We work with mutagenesis in the past, okay? And then also breeders in general working with the entire genome. So more or less you work with the whole species, with the whole plants to make such improvements. So this is kind of becoming very challenging and very time consuming. So this is just an illustration from Chan, published in Annual Review of Plant Biology in 2019, that in general, conventional breeding takes many generations of crossing and selections. Takes about 8 to 10 years, although now with the new technology, you may decrease it into like 4 or 5 years for developing varieties. At the same time, you may also seeing like the narrowing of genetic diversity. There's a potential, there is a linkage drag. That's also quite labor intensive and require a lot of capital investment for this process to occur. So nowadays, as Dr. Pixley also mentioned, that we're now using kind of in the modern plant breeding, we use still using the phenotypic selection data assessment in the field, and combining that information with the DNA sequencing, RNA sequence data, as well as the proteomics data to make us into much better understanding about the traits that we are working on, and to allow us to do like a more precise selection. And at the end, we can deliver the product or the improved cultivars much quicker to the farmers. So we are, for example, been kind of working. My specialization is mostly working on legumes. So in collaboration with colleagues from around the world, we look into different aspects. For example, we have published a number of genome sequence. The first one we did was in chickpeas back in 2013. It was my varieties that was sequenced with colleagues from ICRISAT, from United States, Australia, China, and a number of other countries as well. And also we look at the ecology and genomics, kind of GWAS, environment GWAS on chickpeas, and new collection of wild chickpeas we did in Turkey because of very limited amount of genetic resources available for chickpeas. So the group went back to the origin where chickpea was domesticated and did recollection of those chickpeas, and we characterized the environment where they were grown, and kind of make the association looking at the characteristic of those diversity. So we published that in 2018 in Nature Communications, and just recently in 2019 we published the reference genome for field peas. Field peas in Indonesia known as kacang ercis. This is the species that was used by Gregor Mendel to develop his genetic theory, the genetic science. So only after 154 years after Gregor Mendel used this species, we are able to crack the genome sequence and to understand the structure and variations, and to understand also the legume genome evolutions at the same time. Because field peas is quite big, the genome, about 4.5 billion base pair. So it took us quite a bit of time and effort to get this. But finally we published, and then now we are kind of using all the information from this genomic data that are available into our breeding program to make a even better or bigger progress. So for example, one of the use that we are doing, we are using of those information, genome-wide and QTL analysis. So either biparental or multiparental crosses or germplasm collections. So we create populations and then characterize both and phenotypically in very much detail, and then did all the genotyping and do the association analysis to kind of more or less come up with a gene discovery, what genes are important in affecting the traits, and then how we can modify that genes using the newer techniques such as gene editing to make it even more efficient, or to knock it off, for example, the one that we don't really desire to have such gene to be functioning. So for example, on the corner there's my own plots on chickpeas evaluation nursery, and then we genotyped and we did all kind of analysis. And because I'm at the university, so part of this project quite often is become like a student thesis project. So for example, right now from the collaboration with colleagues from ICRISAT and UC Davis and a number of scientists from Australia as well. So we kind of very much understand globally in chickpeas where all the major QTLs that are associated with abiotic stress across the chromosome of chickpeas. So part of it was published in TAG in 2020. So you can see a number of abiotic stresses and then the distribution across the 8 chromosome in chickpeas. So we have this information very much kind of in hand, and now we're looking at the potential, kind of how much variation between this QTL in the germplasm that are available, and then use that information again into the breeding program. We use this into our selection and into our analysis. So the picture on the left, for example, this is the phenotyping facilities at the University of Adelaide, where we did collaboration with scientists at the University of Adelaide to characterize the water use efficiency and drought tolerance in chickpeas. And we did all the sequencing. So this is part of the project that we're done. And then the information from here is kind of, we use this to make a much faster selections at the early generation from the breeding program. So at the end of the day, probably we would like to sequence on all the genes that are available in chickpeas. So remember that chickpeas, unlike soybean or corn, is a small crop. Only a few people in the world working on chickpeas. So kind of we're a little bit behind if you compare with the big crops like corn, soybean, rice, or wheat. But we kind of we're making progress with all the effort that we are doing. Okay, so another examples that we are doing, probably you may have heard what we call as a genome selection. So in the genome selection now that we have, a lot more SNPs or genetic information are available into the germplasms. So combined with the phenotypic data, evaluation data that we have across the environments or on the specific environments, so that allow us to kind of develop a model using that information. Okay, that's what we call genome selection. We create a model and then using that model to kind of use that model to select the populations or breeding lines that kind of never are not phenotypically evaluated, but we screen them or evaluate them just for the based on the genotypic data only, and then we make selection based on that genotypic data. That's what we call as a GBV or genomic estimated breeding value. So you can see here, for example, the yellow here, part of this graph. The yellow here is the training population. We create the training populations, and then we genotype and phenotype those training populations, and then create the models and applies the model to the breeding materials by genotyping only, calculate the genetic genomic estimate breeding value, and then make selection and continue the process with that selection. So what are important, how to do it? Okay, we set up the training population, did both genotyping and phenotyping, and then using like in the old days, probably everybody familiar with, regress the phenotype on the markers of the genotype, and then use these regression equations to predict the phenotype from markers only in the novel germplasms, and then make selection, make a cross, and repeat again. So after a few generations, you can kind of refine the model and then continue make selection from there. So how many this can be done? So kind of really depending on the linkage disequilibrium, population structure, and the heritability of the trait. So a lot of computing effort here, but this is kind of the one that kind of promising in the breeding program to kind of to make it high precision and shorten the process. Okay, so we applying this in a program on the kind of specific populations, and we see some progress now coming from this application. So this is just an illustration how we doing it. Okay, so early germplasm pool, you have a collection of germplasms. Okay, and then you create the training population, the diverse lines from these populations, consisting of the lines from elite combinations. And then you do both phenotyping and genotyping on these populations. Different from GWAS or QTL, there is no need for QTL mapping or no need for testing a significant markers in this case. So based on these two information, phenotyping and genotyping, you create a genome selection model, and then you also kind of create advanced line, continue, improve, and refine the model. And then you take the breeding lines, untested lines, and you do the genotyping only on this untested line, and calculate the genomic estimated breeding value. You apply the model that you develop, and you make selections, intercross among those selected lines, and then you can test lines at the final stage, and then you can release as varieties. So this is the very much summary of this genomic selection. I'm sure some of you are already familiar with this kind of model. Okay, so a lot of models can be tested with this selection scenario. Okay, so the model can be, the most common one is rich regression blob or G blob. Dimension reduction method, for example, partial least squares, principal component regression, variable selection model with base and kernel or machine learning method. So a lot of model you can test for this genome selection. The challenge with genome selection is that you have a large amount, the matrix is kind of the number of lines that you are testing, usually in the hundreds, in the 500 or maybe 1,000 or a few thousands. But the P, the number of markers, is the one that really huge number, somewhere between, we tested up to 60,000 SNPs data in our model. So that's become a challenge itself when you run such model. So this is the classic paper that was published a number of years ago. In 2001, where Musen kind of put a model, which model the best for the genome selection. So kind of you can see blob genome selection model compared to the least squares, where it's kind of the endpoint is that to compare the correlation between the genomic base breeding value compared to the true breeding value that you evaluated phenotypically. So very much the base A, base B, and blob giving quite high correlations over 0.7 up to like over 0.82. So this is the model that probably you can apply. And this is kind of could be different from trait to traits and population or different genetic structure that you have. This is our own experience in chickpeas, where we tested a number of traits, and then we look at the prediction accuracy with different size of the training populations, from this to flowering trait, this to maturity, the amount of iron content in the seed, which is the kind of important nutritional value in chickpeas, and also Ascochyta blight, SAB here, one of the major disease in chickpeas, seed size, also reflected as 1,000 seed width and yield. So we look at this variability, and then we evaluate it across different environments. So we can see, and then we apply the model, you can see that the correlation we can get really depending on the trait itself. Okay, and then on the right graph here, I'm showing you about what is the kind of optimum, the training population size that you can apply to create the model to make this genome selection. So you can see it ranging from 84 up to over 200, still kind of reliably used for this genomic selection in chickpeas, which is a self-pollinating crop. A number of other techniques that people also use in the breeding, as now we are kind of going into more genomics, and this is the one that been kind of a while ago. This is the GM crop or genetic transformations. So in this case, the source of the gene can be any species. Okay, it's not restricted to the same species. So it kind of really there's no cross barrier with this. So it can be introduced into the genome. This method can be much faster than conventional breeding. And again, some regulations and other techniques, this can be quite expensive to satisfy the regulatory, especially if we go with this route to do the genetic improvement. And then sometimes I was asked quite often, so which one to use? Whether to use GM or whether to use molecular breeding in general. So this is kind of a general rules, kind of a rule of thumb that you may decide. Okay, so in the molecular breeding, the desired trait, the traits that you're looking for must be present in the populations. And then the genetic resources must be available in terms of the genetic data, genomics, and etc. And the plant should be propagated sexually. So you need to make a cross You need to create the recombinations, and then so you select the recombinants using the genetic resource. In GM, gene can come from any source, from any species, even kind of any family, any kingdom. And the GM, genetic resource, not required as long as you have the source of the gene, and plant can be propagated vegetatively. Okay, so that's kind of the option here. Okay, so this is the technique that's been around for a while. Most people are familiar with, and depending on the resources that you have, you can choose one or the other or both. The other technique that just recently coming through is the gene editing and the new breeding technique. So this is the one thing that kind of Canada has not decided kind of what kind of regulation that will go for this gene editing product. But I believe in US they have decision that the gene editing is not going kind of just regular, so it's not regulated as strict as a GM product. So basically what is gene editing is that we make use of DNA break and repair mechanism within the plant cells. Okay, in this case, for example, when plants are exposed to any external stress or anything like that, the plants kind of, there's a DNA breaks and the plants will kind of improve or correct that break and do the repairs. Okay, so this is the mechanisms that naturally occur within the plant. Now we make use of this mechanism by manipulating and by creating the DNA break in the very specific site in the genome. And then the next step is kind of whether you put the kind of template so that the plants will kind of repair based on the template that you inserted. Okay, so basically CRISPR-Cas9 is kind of a cluster regularly interspaced short palindromic repeats. Okay, you create a guide RNAs to direct the enzyme Cas9 to cut the DNA sequence of interest within very specific sites. So once this guide RNA kind of find the matching sequence in the genome, then the Cas9 will cut that part, and then we'll create a double strand break and repair through plant's own mechanism. So this is the things that you can manipulate, people can manipulate with this repair system, whether you want to create a stop codon, you want to kind of create a mutation or a lot of things that people can do with this mechanism. Okay, so for example, from that double strand break from the Cas9, you see there's the possibly non-homologous and joining, random insertion or deletion, gene knockout, or you can have the homology direct repair where you can have like a precise editing from this process. And also now people improving not only Cas9, there are a number of other enzymes that are recently come into play that people can use in very much different ways of doing the gene editing. So this is really an efficient technology that targeted this is the technology will enable customized solution for crop production challenges in many parts of the world. So for example, it is really kind of site-specific DNA integration, pest-resistant plants, enhanced nutrition, disease resistance, and drought tolerance, viral resistance, tasty vegetables even, genome engineering. So a lot of applications kind of really the one thing that will come from this technology. Keep in mind that this is much different than the GM because in gene editing, kind of just tweaking the genes or part of the DNA in that own species. So you're not transferring, you're not introducing any other segment of DNA from other species, but within that species, within that same plant, you're just tweaking it, you create a modification within that same plant. Okay, so this is kind of the generating editing plants. You really need to know the target sequence, the target gene that you want to edit. Okay, so and then you design an experiment. There's a number of tools that are available, but the first thing really you need to know the sequence, which part that you want to modify, or you want to knock out, or you want to add, modify the changes. Okay, so you need the sequence data, and then you need to know the gene structure. Yeah, and then you make a target which part that you want to edit. And then you need to have the plant transformation and regeneration systems. And then from there, you will do the molecular analysis to confirm there is an intended mutation happen in the target genes, and there is no other unintended integration in different part of the genome. And then finally, you get the final product with improved traits. So number of crops, this can be done easily. Number of crops, for example, in legume, the plant transformation and regeneration is quite challenging, especially the plant regenerations. It's still recalcitrant or very difficult to develop an intact plants from the callus or from the culture in legume crops. Okay, so CRISPR-Cas has really broad applicability across the crops and traits, across different genome size, ranging from 0.5 up to like 16 billion base pair can be done in any crop. Okay, and the process also allowed to have multiplexing. Okay, meaning that not just one gene, but more than one gene you can edit at the same time. For example, here in maize, people have done as published in 2015 by Ligulus gene region, male fertility, and then 2 male fertility genes being edited at the same time in maize. Okay, a number of other applications. For example, this has been published in 2018 in PNAS, where the group from Purdue modified or editing the abscisic acid receptor, the pyrrobactin resistant like 1 or PYL1, and then changing the plant morphology, changing the plant adaptation to stress in rice, and the end product from this editing in rice by Meow is increasing the yield potential up to 33%. So in a way, if you look at the gain that people work on hybrid rice, it's almost like as much as gain people get from hybrid rice by doing this editing, which is much quicker process than creating the hybrid rice in the process. A number of other applications, clonal production of hybrid rice. So this I just give you here a number of publications that have been successfully reported of gene editing for different purposes. This is, for example, for yield, where allow the plants by modifying mime genes, okay, so that the plants, instead of doing the hybrid, now it can do the clonal reproductions in rice. A number of other examples, for example, by changing the plant morphology as approved rapid domestication in tomatoes by modifying or by editing a number of different genes associated by architecture, by the LN sensitivity, by the fruit size, and increased vitamin C levels in tomatoes. There's been kind of 2 papers published in Nature Biotechnology in recent years. And also low GI rice for diabetic by kind of modifying the SBE, start branching enzyme genes in the process. So that's already been published in 2017 in Frontiers in Plant Science. Also a number of resistance to bacteria by modifying the susceptible gene editing, sweet genes in rice. Okay, they also been reported to have that improvement in rice. Okay, also to plant pests, for example, in rice, okay, stress tolerance, and also to brown plant hopper resistant in rice. Okay, it's a Nature Plant in 2018 was published. Okay, so we kind of put a review as I'm working on legumes, what are available in legume and where we are now in legume. So we published this just recently in May 2021. So I was one of the co-authors in this paper. Okay, so feel free if you can access or you cannot access, you are interested, you can email me. I will send you a copy of this paper. This is very much a kind of representing the information on legume where we are now in terms of the application of CRISPR-Cas9 technology for this group of species. On our own experiment, we kind of doing a proof of concept in chickpeas. We The first one, kind of the easy one, perhaps an easy target, where we try to modify to increase the beta-carotene content in chickpea. Chickpea already has a kind of significant amount of beta-carotene, but can we increase? So we know the gene sequence, so the PDS, pathway indesaturase, and also a number of other genes in the chickpea that we modify. Okay, so we create a guide RNA targeting that PDS gene. We did the protoplast base using the PEG electroporation delivery system, and we created the kind of a callus from this transformation from the edited genes. And okay, so and we know that we look at we did kind of a number of different guide RNAs, so up to 5 we did. So the one that was successful is on the guide number 3, which is on the exon number 10 of the pathway indesaturase gene, PDS gene in chickpea. So you can see there's insertion of A in this gene, and then you can see at the bottom there the callus or the materials coming from this editing process, kind of a lack of development of associated with a chloroplast or carotenoid to look like pale. Now the challenge for us is that to develop from this callus, from this plantlets going into an intact plants, that's really the challenge in most of the legumes. So we did also some gene editing with the herbicide tolerance, okay, so with metribuzin tolerance, which is now ongoing. So really, this genome editing and genome breeding, base breeding, over breeders, unprecedented ability to quickly develop, improve plant varieties, which can stabilize and increase yield despite the changing environment, withstand the pest, favor crop input, maximize the resource use efficiency. And at the end, we hope that within that 30 years, we can increase global food productions to meet the population needs. Okay, so from here, just throwing an idea, so what will look in the future in the 50 to 10 years, beyond just an improvement of plant traits or new varieties in the plant breeding or in crop improvement. So perhaps with a much better understanding of the genome and the biology and the physiological process, there could be a synthetic species that we kind of assemble all the information and create a new species. Okay? Or probably on the engineering part, there could be a photosynthetic machines that are converting carbon, hydrogen, oxygens into organic compound. So the bottom line I would like to say here, we as human beings never underestimate human creativity and capability. You never know what people's creativity is. So we always being creative as human being and the capability with God willing, we may create a different thing that will be very useful for human being. So with that, this is my group, former students, former PhD students, master students, postdoc, and some of them kind of back to their own countries and working in different places. So I'd like to thank them for all the work that we're doing together. And thank you again for your attention. And I'll be happy to answer questions if there's any from the audience. Thank you very much.
Thank you, Prof. Benjamin. Let I check the question from chat. There are several questions, Prof. Benjamin. The first one is from Ibu Nuning. Dr. Taan, breeding is one of the lines in production system, plays an important role in providing superior cultivars. With the passage of time, whether the more advanced breeding becomes a guarantee of production efficiency? Please, Dr. Taan.
Okay. So I'm trying to read him because at some point Prof. Suyamto voice kind of being cut on my end here, so I'm reading again. So breeding is one of the lines of product in production system play important role providing super cultivar. So again, breeding in general rule of thumb is kind of 50% of the improvement in productivity coming from breeding. So the other 50% is coming from crop management. So these two together, the breeding, which we deliver the potential genetics for crops, must be accompanied with good practice or good management. So all this has to come together to get a guarantee of production efficiency. With the breeding, for example, production efficiency can come with the increased yield stability. And the simple example I can give you with the production efficiency, for example, chickpea crop in the old varieties that people in Saskatchewan in Canada grow, they need to spray fungicide up to 5 times in a year during the plant growing cycle. Now with breeding, with improved disease resistance, it's not immune, still some susceptibility, but now the growers only apply once or twice of the fungicide. So that's already production efficiency that coming into that while maintaining the yield. So that's a kind of a simple example I always give to the farmers. So this is a kind of a real simple that you can so it's kind of a combination, the genetics. So we continue to develop the genetic materials, because without the genetic materials, no matter how good the management, if the genetic potential is not there, then you may not see the result. Okay, terima kasih. Thank you, Bu Nuning, for the question. That's very important questions.
Thank you, Pak. The second question is from Dr. Willy. Dr. Taan, thank you for the enlightening presentation. What are the challenges for implementing genomic prediction in legumes crops? Thanks. Please.
Thank you again, Pak Willy. So in legume crops, what is really the challenge in implementing the genomic prediction in legume is that creating kind of the most appropriate training populations. So that's really the challenge for us here. That kind of can be adapted or can be evaluated in one environment. So because the genetic resource is quite limited for some crops in legume, okay, so creating that sizable training populations, that's really still a challenge for us. So we kind of in a way create different models, okay, based on the best we have right now when we develop genomic prediction, using the training populations that uses all the foundation lines that we use in the program. So the old varieties from 1940s, 1950s, the germplasm base that are available, and then I know they've been used in crosses in different part of the world. So that's the one thing that kind of really the model. So in a way, phenotyping of those training is the one that challenging. Genotyping is the easy part these days. Genotyping is the easiest part. If you have funding, you have resource, or even you have machine, you can generate unlimited kind of a large amount of genotypic data. But phenotyping, for example, if you do, especially when the process involves wet chemistry, for example, when we do carotenoid, beta-carotene, for example, you need to use SPLCs. Then you need to do kind of replications, different locations. So that's becoming very expensive. That's become kind of the limit when we do the implementation. But the good side is that once we have a good model based on that phenotypic and genotypic data, then the application really going to be very much useful. So I hope that answer your question, Pak Willy. Thank you very much. I hope I will see you again someday, sometime, maybe in Indonesia or in Bogor.
Well, thank you, Pak Bunyamin. I would like to give the participant here to raise the question directly to Pak Prof. Bunyamin. Is there any question from here, from this room? No. Pass. It means that no question. Pak Bunyamin, thank you very much for your excellent presentation. I think we hope you can come again to Indonesia anytime after pandemic, I hope. Yes, and I think
Of course.
yes, what you represented is very, very excellent, and I hope that our breeder can learn, can take the lesson learned from Pak Pujamin to increase the effectivity and the faster breeding process in Indonesia. With this, I would like to close this session. Thank you very much for your participation. Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih Prof. Suyanto.
Honorable speakers and participants, ladies and gentlemen, for the next session, we are honored to have the next speaker, Dr. Kirti S. Rathore, Professor of Texas A&M University, United States of America.
Presenting his paper, Genetic Improvement in Dicot and Monocot Crops, and the chairman of this session is Professor Yunus Musa from Hasanuddin University.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for MC allowing me as a moderator in these sessions. Welcome, ladies and gentlemen, in the second international conference for today. Now we are leading to the next invited speaker. He is Professor Rathore. But before that, allowing me to read some of his CV, and then let me choose the best or the more relevant in line with this international conference. Professor Rathore, educations in Rajasthan University, India in 1973. And then his master's in Gujarat University, India also in 1976. And then his doctorate in Imperial College, University of London, UK in 1981. Professor Rathore, really a professional expert in his field. And then from his experience, let me read some of his. In 2012, he is as a professor of Department of Soil Science and Crop Sciences at Texas A&M University. In 1991 to 1995, as a research scientist at the Department of Botany and Plant Pathology at Purdue University, and in 1982 to 1984, as a postdoctoral associate at the Pure and Applied Biology at Imperial College, London. His significant accomplishment and awards is, let me read three of these: established efficient tissue culture transformation system for cotton, sorghum, rice, potato, and tomato. And also conceived and developed ultra-low gaussival cotton seed. And also recipient of 9 US patents, 2 patents pending yet. Professional activities including as associate editor, as a review of panel members, and also trained international visiting scientists. As invited presentations, he has selected peer-reviewed publications including first, ultra-low gossypol cotton seeds, a selected gene. And then also rapid recovery and characterizations of transformants following agrobacterium mediated tDNA transfer to sorghum. Also, a two-dimensional vibrating probe study of currents around lateral roots of Rapanus sativus developing in culture. More of that, he published so many books, but read me some of them. That is, a cotton Gossypium hirsutum in methods in molecular biology. Although cotton, again, in biotechnology, in agriculture and forestry. And the last one that I would like to say, that he is also, again, published a book in cotton methods in molecular biology, agrobacterium protocols. I am very sorry for Professor Rator that I cannot read all this very long CV, but that's the most important ones. Ladies and gentlemen, now we have Professor Rator as a very reputable scientist in his field. Good morning, Professor.
Good morning. Can you all hear me?
Thank you for being with us, and then time is yours, please.
Okay. I would like to share my slides. Okay. So can somebody, okay, now we got it. Okay, so everyone can see.
Yes.
Okay, well, thank you very much, Professor Musa, for the introduction. And I'll start off by thanking all the organizers for this invitation to give a presentation of some of the work that I've been doing at Texas A&M since 1995. Now, my goal is to introduce the audience to diversity of the traits that can be engineered using either transgenic technology or gene editing technologies. The previous two speakers have done a really good job of providing you with the graphics as to how these technologies work, so it kind of cuts down on the work that I have to do. So what I will do is to give you some examples from my own lab. There are 2 examples from sorghum and 5 examples from cotton that will illustrate that even a public institution laboratory can carry out these kind of, or can create this sort of transgenic traits. And then at the end, I'll give you a couple of examples of gene-edited traits that we have created in my lab. So with that, I will start my slides. So again, because this is a very diverse audience, I would give you some basics and then talk about some of the examples. So genetic engineering, what is genetic engineering? Well, one form of genetic engineering, which has been around for about 25 years, is called transgenic creation or transgenic plants or genetically modified organism or GMOs. And there are at roughly three kinds of genetically modified plants. One is created by overexpression of a native or a foreign gene. The examples of those you probably all have heard. One is golden rice, another say BT cotton or BT maize. Another way to genetically engineer is to silence a native gene using either antisense technology or RNAi. And one example is creation of low gossypol cottonseed. Now this work was done in my lab, and I will go into much more detail explaining what we did and how we did this and how this trait can be very useful in terms of global food security. The third type is when you express a double-stranded RNA from a pathogen or a pest to kind of address this problem of pest or create pest resistance or pathogen resistance. An example of that is maize rootworm resistance. Now, another form of genetic engineering involves gene-edited plants. Again, you have seen the graphics and everything. There are three different methods to create gene editing. I won't go into detail of any of those, only to say that the most recent technology is the CRISPR-Cas9, and this is what we have used, and most labs these days are using this technology. The previous speaker gave very good graphics about how this thing works. It's a site-specific endonuclease that creates a double-stranded break in the DNA, and the cell's repair mechanism comes into play. The most common one is the non-homologous enjoining. And while repairing with this particular mechanism, you create mutations because of the nucleotide insertion, deletion, or substitution. The other one is a little more rare, but it's called homology-directed repair. And it's much more precise, and you get very subtle desired mutation. However, this requires a DNA repair template. Now, this is not as common as this one. So I'll give you a couple of examples from my lab. So now, first I will talk about transgenic traits based on overexpression of a native or foreign gene. So when you want to create a transgenic plant, normally you basically need your gene that you want to introduce into your species of interest. It has to be under the control of a promoter, and then you need to have a terminator. With this gene of interest, you also need to introduce a selectable marker gene so that you can select the cells that have been transformed. So I won't go into detail. This is there are times when you require some other components also, and we don't have time to go into all of those. So before I start to show you my examples, the non-transgenic plants, I am referring to them in some slides as wild type, meaning the parental line that was transformed. So that's control, non-transgenic plants, wild type or WT. Sometimes we use the null segregants, and they'll be either labeled as null segregants or NS, or I'll simply label them as control. So all of them are non-transgenic plants. the transgenic plants will have some sort of number. So first two examples are from sorghum. The first one we have done is to create some sorghum plants that are resistant to herbicide Basta or Liberty. And the second one deals with improving nitrogen use efficiency. So as you all know, sorghum is used as food, feed, and also as fuel to make some alcohol that you can utilize as fuel. So again, won't go into too much detail. These examples, most of them I'll just show in the form of some slides or pictures. So this is the example of sorghum plants that we had created that are resistant to herbicide called Basta or Liberty. And this sorghum plant were created by introducing a gene we call bar gene, which came from this bacteria called Streptomyces hygroscopicus. And here are the control plants, non-transgenic plants, sorghum, and they were sprayed with Basta, and after about a week, they're all dead. Here are transgenic sorghum plants that have the bar gene, and they were sprayed with the same herbicide, and they are resistant to Basta. And we have already published that, and you can look that up, those papers. Then another project we have done is to improve nitrogen use efficiency. As you all know, nitrogen is an important macronutrient. It's critical for crop productivity, and the demand for nitrogen is growing every year. But unfortunately, plant only takes up about 30 to 50% of the nitrogen that we supply. The remainder contaminates the soil, water, and air. And so it creates a lot of pollution and also is responsible for some of the greenhouse gases. So we made a construct where we utilized glutamine synthetase gene from sorghum. The promoter here we used was maize ubiquitin promoter, and we put this construct into sorghum, and here is one of the results. So under optimal nitrogen conditions, there is a big increase in shoot biomass in the transgenic lines, so almost double the shoot biomass compared to the non-transgenic or null segregant. As shown in this graph. Here is a picture of those plants. So here is the null segregant or non-transgenic sorghum plants. You're seeing just one tiller, whereas the transgenic line creates multiple tillers. So there is accelerated formation of secondary tillers in this. And so you end up with higher shoot biomass as well as higher number of seeds from these plants. And so this is shown in this table, although there is the primary tiller, the grain number in the transgenic is a little bit low compared to the control. However, this transgenic plant produces secondary tillers, so you get many more seeds, and by this time there are no secondary tillers are produced in the non-transgenic ones. So you're increasing the number of seeds as well as the shoot biomass. And that's basically this conclusion tells you that. So those are the two examples from sorghum. We've done some other work on sorghum. I would not go today into the details of that. But we have done much more extensive work on cotton. Cotton is an important crop in Texas, and U.S. is #3 producer of cotton. So it's an important crop for U.S. as well as Texas. Now, first we had to work out a reliable and efficient transformation system. I say transformation system meaning or ability to introduce a gene into cotton cells and then get a fertile plant back from those cells. And a efficient and reliable transformation is the foundation for transgene overexpression or gene silencing and genome editing. And I'll give you some examples from each of those three types of genetic engineering. So this is just to give you some idea as to what is involved in transformation of cotton. So you start off transformation of cotton. Here is a leaf disc or a cotyledon disc, and we have utilized this green fluorescent protein, and that's to transform. You can visualize the transformation because the cells that are transformed are going to appear green under fluorescence light, or will under UV light, they'll fluoresce green. So anyway, starting from this day to getting some plants in soil, you have to go through this tissue culture process, and after extensive tissue culture, we get somatic embryos that grow into little bitty plantlets, and then we can go to the soil. So going from this stage to this stage can take anywhere between 12 to sometimes 14 months. So the first example I want to show you where we have introduced an osmotin gene from tobacco into cotton cells and generated transgenic cotton plants. So this is the cotton plants. These are two different transgenic lines. Here is wild type control, non-transgenic. We grew these plants in greenhouse, and we withheld watering for about 9 days. And as you can see, after 9 days of not watering these plants, they are severely wilted, whereas these two transgenic lines appear fairly healthy. There are some wilting in the cotyledons, but the leaves don't show this wilting. So that was very good results. So we carried out a much longer term experiment where we took the plants all the way to maturity, and these were done in bigger pots. We applied drought stress during 3 times of the life of the plant, and these are the times when we applied the drought stress. After first 10 days of drought stress, this is what the plants look like. So here is wild type or non-transgenic cotton plants. Here is a transgenic line, and as you can see, the plants are wilted here. Plants look fairly healthy in the transgenic lines. So we took these plants all the way to maturity. So we have here comparison of well-watered plants, and we are looking at the yield of seed and the lint or the fiber. Under drought conditions, we get much higher yield of fiber or the lint compared to in the transgenic lines compared to the non-transgenic. So we got a pretty interesting results in the greenhouse. So that's one example. Second example I will show you deals with creating cotton plants that are resistant to certain diseases, fungal diseases mainly. So this example, we have utilized an endochitinase gene from another fungus called Trichoderma virens. And Trichoderma virens actually is a pathogen of other fungi. Now, cotton plant as well as several other species suffer from infection from Rhizoctonia solani. In cotton, it causes a disease called the seedling disease. And if the soil contains Rhizoctonia solani, the seeds either do not germinate or die soon after germination. So we took this gene, endochitinase gene from Trichoderma virens, put that into cotton, and created these transgenic line. One of the transgenic line is 135-4. The soil here contains Rhizoctonia solani, and these lanes here are transgenic seedlings. they germinate nicely and do not show any effect of the fungus disease, whereas the non-transgenic seedlings die soon after germination. Now, this particular gene gave us resistance to only a couple of fungal pathogens. So we did another experiment where we actually took NPR1 gene from Arabidopsis and put that into cotton. With this gene, we actually got resistance to multiple pathogens of cotton. So we tested Rhizoctonia solani, Thylabiopsis, Alternaria, Fusarium oxysporum, and Verticillium. So here are the results. So here is Rhizoctonia solani. So wild type non-transgenic seedlings, as you can see that they are severely infected, whereas the transgenic line, the seedlings are fairly healthy. Thylabiopsis, which causes black root rot disease. Transgenic line shows the non-transgenic plants are stunted here, wild type. You can see the disease right here on the root. And so both the roots are stunted, the shoots are stunted, whereas the transgenic lines, there are 3 types of transgenic lines here, and the root, they do get infected, but they're able to overcome the disease, and they do grow much more vigorously compared to the non-transgenic seedlings. And same with the shoots, they're much more healthy compared to the wild type. So that's Thylabiopsis. The same gene also confers resistance to a foliar disease called Alternaria alternata. So this was done in petri dishes. So here is a transgenic line, fairly healthy after 7 days of infection, whereas this is wild type or the non-transgenic line infected and shows severe infection. We got some tolerance to Fusarium also. So this is wild type and this is transgenic line. Similarly, we got some tolerance to Verticillium, non-defoliating isolate of Verticillium. And so again, this is wild type and transgenic line. So we got a very broad spectrum resistance to several fungal pathogens. Another example deals with the use of a gene we call PTXD gene. It's isolated from a bacteria called Pseudomonas sturtzeri. It codes for an enzyme called phosphide dehydrogenase. Now this enzyme can catalyze a reaction in which phosphite is converted to phosphate. Now this work we've done in collaboration with 2 Mexican scientists. Now this gene, or the use of this gene, is to give us an alternative technology to control weeds, and I'll give you a couple of examples as to how effective this technology is. Okay, before I go to those slides, I just want to say that as you all know, plants can utilize only phosphate form of phosphorus for nutrition. If you provide them with phosphite or PO3, plants will absorb it, but they cannot utilize this form of phosphorus. But if you have this gene, PTXD, which encodes phosphide dehydrogenase, you can convert PO3 to PO4. So we took this gene, introduced that into cotton plants, and the results are shown in this slide here. So we did some computation experiments. So one of the limitations of this technology is that the soil has to be low in phosphorus, otherwise it will not work. So this soil only had about 18 parts per million of phosphorus. And this was a competition experiment. So we had 6 cotton plants in this tray, and we also had some amaranthus seed, Palmer amaranth seeds in this soil. And after about 4 weeks, you can see the results. So here are wild type or the non-transgenic cotton plants in competition with the weed, Palmer amaranth. Here is a transgenic line in competition with the weed, and the weeds grow so much that you cannot even see the cotton plants. Here, the fertilization was done with phosphate. In this case here, the fertilization was done with phosphite. Now, as you can see, both the cotton plants and the weeds are really stunted because they cannot utilize phosphite, whereas the cotton plants that have the PTXD gene are able to grow and outcompete the weeds, and so they can grow fairly normally. So that's one example. Another experiment that we did was on a different kind of soil, and this soil had only 10 parts per million of phosphorus. And before we conducted this experiment, we did not know that this soil naturally contained another weed that's called the broadleaf signal grass. So as you see this grass growing here, that's a broadleaf signal grass that was naturally present in this soil. But we also had introduced the weed, the Palmer amaranth, in this tray, and there were 6 cotton plants. So again, here, wild type cotton plants against the weeds. Here is a transgenic line against the weeds. The fertilization was done with phosphate, and the weeds outgrow the cotton plants, and you can barely see the cotton plants. When you fertilize with phosphite, in this case, both cotton plants, the non-transgenic cotton plants, and the weeds are stunted, whereas transgenic cotton plants grow nicely, whereas the weeds are stunted. So this technology, and we are doing some experiments under field conditions to see how well this technology works under field conditions. The only limitation is that the soil has to be low in phosphorus. So those are some examples where we are overexpressing either a native gene or a foreign gene into cotton or sorghum. The next example I'm going to give you deals with silencing of a native gene in plants. And we use a technology called RNA interference or RNAi. And simply what you have here is inverted repeat of a target gene that you want to silence. And this is in the middle, we put an intron. So when this gene with this construct is transcribed, you get this hairpin-like structure. The RNA will form a hairpin-like structure. There is a mechanism called RNA interference that comes into play and that silences the target gene. And this is the one I'll go into a little more details. So this is a trait that we created in my lab, and we call that ultra-low gossypol cottonseed. It's an RNAi-mediated trait, and this is the one that I have spent almost 25 years working with. So why do this work? Well, those of you who know cotton would know that cotton plant produces more seed than it produces fiber in terms of weight. So in the year 2014, worldwide we produced 26 million tons of fiber. The same year we produced 47 million tons of cotton seed. Now cotton seed has about 22-23% protein. It's a relatively good quality protein. So what that means is that this amount of cotton seed contains almost 10 million tons of protein. Now, a human being requires about 50 grams of protein per day to survive. If we could use all this protein directly for human nutrition, we can meet basic protein requirements of almost 500 million people per year. Now, we cannot utilize this cottonseed directly for human nutrition or even as feed for some of the animals because of the presence of this terpenoid called gossypol. And I'll give you some details about this gossypol in a minute. But by the way, this is a big pile of cottonseed about 400 miles from where I am. It's a big cotton growing area, and this is just cottonseed pile next to an oil crushing plant, and this is all going to be processed into oil. So what is gossypol? So this is the structure of gossypol. Now, if you cut open a cottonseed, you'll see these black dots that you see here, and these are glands, lysogenous glands, and these contain gossypol. So, as I said, the seed contains about 22-23% protein. It's relatively good quality protein. Now, because of the gossypol in the glands, we cannot utilize this seed directly for human consumption or even for animal feed. Most animals cannot tolerate gossypol, except for the cows. Cows can tolerate certain amount of gossypol. So gossypol is toxic to a lot of monogastric animals and human beings. It damages heart and liver. But gossypol is typically removed during the processing of cottonseed to obtain edible oil, and we all do consume cottonseed oil. So, as I said, cottonseed oil is prepared by a lot of processing, and the processing removes gossypol, and after that it's safe for people to eat, and we do consume a lot of cottonseed oil in this country and many other countries. The rest of the meal, which has a lot of protein, is either is fed to the cows either before or after crushing. But some of these animals cannot utilize cottonseed as feed because they're all monogastric animals. Now, what's wrong with that? We do not throw away the cottonseed. We're feeding it to the cows, but cows are extremely inefficient in converting feed protein into meat protein. If you want to get just milk, it's not too bad. But if you want to get meat, then the protein conversion ratio for cows is very high. It's 20. What it means is that you have to provide 20 pounds of feed protein to 1 pound to get 1 pound of beef protein. So that's the protein conversion ratio for cows. Compare that with these other animals. So protein conversion ratio, or PCR, for cows is 20. For pigs is 5.7, chicken 4.7. So chicken technically is 5 times more efficient compared to cows. Eggs is the most efficient way to convert feed protein into edible animal protein. Some of the fish here is also very efficient in converting feed protein into animal protein. Now, gossypol and some other related terpenoids are also present in the leaf, in flowers, in the ball, bracts, stem, and so on. You can see these glands. So gossypol and some other related terpenoids are present, and their function is to protect the cotton plant from insects and some diseases. Okay, so what is a desired trait? What we want is to have the seeds, cotton seeds, to have very low or no gossypol, at least have gossypol below the level of 450 parts per million. A normal cotton seed contains gossypol at a level of 10,000 parts per million, but if we can bring down the level of gossypol to 450 or lower, FDA in this country considers it safe even for human consumption. But we want to maintain gossypol and the related terpenoids in all other parts of the cotton plant so that the plant can protect itself from insects and diseases. So this slide shows gossypol biosynthesis pathway. So one thing to remember here is that the glands in the cotton seed and the flower petals contain just gossypol. The glands in the green parts of the cotton plant contain not only gossypol, but hemigossypolone and the heliosides. All these terpenoids are necessary for to protect the cotton plant against insects. In fact, these compounds have much higher insecticidal activity. Now, they all come from the same biosynthetic pathway. Here is a very important step. It's conversion of farnesyl diphosphate to delta-cadnine. This is driven by an enzyme called delta-cadnine synthase. So we thought, okay, if we can block this step using the RNAi technology, but block it only in the seed, then maybe we can eliminate gossypol in the seed, but the rest of the plant will still contain normal levels of gossypol and terpenoids. And that's exactly what we did. We isolated a promoter from cotton, which is active only in the cotton seed. And we tested that activity using this reporter gene. So we call this promoter alpha globulin promoter AGP. We introduced this promoter driving a reporter gene into cotton, and this promoter becomes active at 16-day post-anthesis. This reporter gene, when it's active, you can see that this color of the embryo gets blue. So that means the promoter is active in 16-day post-anthesis, but the activity increases as the embryo grows, and by day 25, the promoter is active throughout the embryo. The other thing, we wanted to make sure that this promoter is active only in the seed, and this graph shows that the promoter is not active in any of the other organs, it's only active in the seed. So we knew that we had a good promoter which becomes active at about 16 days post-anthesis, but the gossypol production does not start until day 25. So we knew we had a good promoter. Again, so we made a hairpin construct, just the way I described earlier, and introduced that into cotton. Here is our first result. So this is gossypol values in cotton seeds, individual cotton seeds. So we took 10 seeds from wild type or non-transgenic cotton plant, and these are the gossypol values. So it's about 10 micrograms per milligram tissue, or 10,000 parts per million. That's about the average. Here are two transgenic lines, or RNAi lines, line number 2 and line number 32. As you can see that some seeds that are very, very low in gossypol. Now, these are the seeds that had the RNAi construct. These are seeds that don't have the RNAi construct because they are null segregants. So the seeds are segregating according to Mendelian principles. So we carry out HPLC analysis to detect gossypol. So in the non-transgenic seed, the HPLC peak looks like this. In the transgenic RNAi lines in the seeds, we can barely see the gossypol peak. So it shows that the gossypol is reduced. So gossypol is reduced in the seeds. What about other parts of the plant? What about the leaf? So we looked at the leaves. So again, we have the non-transgenic wild type, the two transgenic line number 2 and line number 32. Again, if you remember that green parts of the plant contain not just gossypol, but also hemigossypolone and heliosides. And as you can see that the gossypol is not reduced in the transgenic lines, or same thing with the other two types of terpenoids. What about other parts of the cotton plant? So we also looked at the terminal bud, floral bud, bracts tissue, flower petals, ball, and the root. And again, the red bar here is non-transgenic plant, and the two green bars are transgenic lines. Again, we looked at all the terpenoids that are present, gossypol, hemigossypolone, and heliosides. And as you can see that we do not see reduction in the terpenoids in the transgenic lines. So what that result shows is that the low seed gossypol trait is completely tissue specific. This is another line, line number 81, and this is what the seeds will look like. So we still have the glands, but the glands have very little gossypol. In this case, we only have about 190 parts per million compared to this control seeds, which is almost 10,000 parts per million gossypol. So after that, we conducted field trials for 3 years. We were looking at this trait and see whether it's stable under field conditions, and we also looked at the specificity and the yield and so on. So this is the results for 3 years of field trial done right here at Texas A&M field. Again, red bar is non-transgenic and the green bars are the transgenic lines. So even under field conditions over 3 years, we see that the trait is stable and is stable under field conditions. This is just, we looked at all the other parts of the cotton plant, and basically what we are seeing here is that the leaf and flowers and so on have the same level of gossypol and other terpenoids in the transgenic lines compared to the non-transgenic. So we carried out, we thought this trait is extremely important, so we took it all the way through the regulatory agencies, meaning because it's transgenic, we had to get permission from the government agency before we can grow these cotton plants in the field, or the farmers can grow it in the field. So we had done the science in 2006, but after that we had to do a lot more work to get the deregulation. So we had to generate hundreds of additional lines. We had to do a lot of molecular studies, biochemical studies, inheritance studies. We had to do agronomic performance trial. Now these trials were done by different farmers in 3 different states. We carried out 8 trials over 2 years. We did seed composition analysis, fiber quality analysis, and finally we prepared our application, submitted to FDA and USDA in 2017. USDA gave us deregulated this one event in 2018. FDA gave it approval in 2019. So this is now deregulated and has been approved by FDA. So basically, we've taken the gossypol levels down from about 10,000 parts per million to about 350 parts per million, way below what FDA considers safe for human consumption. The important thing is that the flowers and the leaves and other parts contain the same level of gossypol for protection. And we have just published this review that lays out the whole system. So what can we do with this? Well, now we can take this cotton seed and certainly feed it to the poultry or the pigs. Aquaculture industry is growing rapidly around the world, and we can use this as a feed for aquaculture industry. I believe Indonesia gets almost 1/3 of its fish from aquaculture. Or we can utilize this seed as a source of protein directly for human nutrition in this form. Very quickly, I'll just show you some graphs here that show you what the need is for feed for poultry industry. take the example of India. This is population growth in India from 1961 to 2017, and this is the poultry growth of poultry. So poultry has grown 51-fold from 1961 to 2017. During the same period in China, the poultry production had grown by almost 29-fold. Similarly, we see a big growth in egg production in India and China. Similarly, aquaculture is growing around the world because we're not getting enough fish in the oceans and the rivers and the lakes, so more and more of our fish is coming from aquaculture. So aquaculture industry is also looking for some alternative source of protein. And in fact, we have done some shrimp feeding studies as well as fish feeding studies with our cottonseed. And both the studies found that our cottonseed was good enough for shrimp and the fish. I'll skip this slide. Maybe somebody is going to probably ask me this question, so I'll skip that. And I'll move on to couple of examples from my lab, which involve CRISPR-Cas9 system for gene editing. Now, the previous two speakers talked about creating useful traits using the CRISPR-Cas9 technology, but CRISPR-Cas9 technology also very powerful technology to study the function of a gene. And I'll show you an example of that in cotton. So as I showed you this slide earlier, that the cotton plant contains these glands in all parts of the cotton plant. And you see these glands. Now, we wanted to know what is the gene responsible for making the glands. We did a lot of other studies. We narrowed down the choice to about 10 genes that look like they may be involved in making the glands in cotton, but that's still a lot of genes. Now, how do we confirm that this gene is the master gene responsible for making the glands in cotton? So there was we kind of narrowed down the choice to two genes. We call them CGF2 and CGF3 gene. We sequenced the gene CGF and CGF3 genes, and this is the sequence of CGF3 genes shown in the A genome and D genome. By the way, cotton is a tetraploid, so it'll have 4 copies of the same gene, 4 alleles. So the question was, is it the CGF2 or the CGF3 gene that is the master gene that is responsible for making glands in the cotton plant? And so we did CRISPR-Cas9 mediated knockout of CGF2 and CGF3 gene. So this is non-transgenic control. You can see these glands in all parts of the cotton plant, as you are seeing here. When we knock out the CGF2 gene, we can see some effect on the glands. You can see the glands are much smaller in size, as you can see very clearly here and here also, but they are not totally eliminated. But if you knock out CGF3 gene using the CRISPR-Cas9 system, we totally eliminate glands. What that shows is, and this is another much higher magnification picture of the same plants. So here is glands, normal glands. This is CGF2 knockout, and this is CGF3 knockout. There are no glands at all in CGF3 knockout. And we confirm what the mutations were, and so we were able to confirm the function of CGF3 in the formation of glands. So this technology is a very powerful technology, and you can create a useful trait, but also you can figure out a function of a gene. And the simplest way to figure out a function of a gene is to precisely knock out that gene and see what happens to the plant. this is another example which comes from potato. In fact, we took this picture just last week. So our aim was to, this is in potato, our aim was to eliminate amylose from the potato starch. So as you all know, that starch is composed of amylose and amylopectin. And our goal here was to eliminate amylose from starch, potato starch. Once you do that, starch has many industrial uses. So that's what we wanted, and that was our goal. And so when you take normal starch, which contains both amylose and amylopectin, you stain it with iodine, you see this blue color. So these are starch granules, all stained blue with iodine. But if you eliminate amylose completely, the starch granules will stain red, as you see here. So this is our genome-edited amylose-free potato done with CRISPR. So this is a trait that we have introduced into potato plant using the CRISPR-Cas9 technology. It obviously has some limitations. Single copy genes in diploids are easier to knock out compared to tetraploids, although our cotton and potato examples show that you can handle tetraploids, and you have to do more work, but you can do tetraploid and hexaploid, but that's one of the limitations. Now, I still feel that traditional genetic engineering is still relevant for traits such as BT cotton or BT maize, or there are other BT traits as well. You cannot use, you cannot bring about those kind of traits with gene editing technology. Also, certain traits, just such as the ultra-low gossypol cottonseed trait, are not possible with genome editing technology, because you can knock out a gene, but that will eliminate you cannot make it tissue specific. And we did that by eliminating the glands, but we eliminated the glands throughout the cotton plant. But the idea is to eliminate, say, gossypol only in the seed. That is not possible using the gene editing technology. So normal, traditional transgenic technologies are still useful. Now, I don't know how much time do I have. Okay, I'm getting pretty close to my end. But I'll just briefly say that we already are dealing with all kinds of mutations, and transgenic technology, and specifically gene editing technology, is no different, no more dangerous compared to the older mutation technologies that we have been using for a long time. Nature already carries out a lot of genetic modifications by itself. And at least in the US, we have 25 years of history of safe use of GMOs, and they undergo intense scrutiny by the regulatory agencies. So this is why it took us 25 years to get permission to, from start to finish, to get permission to be able to grow these low gossypol cottonseed in the field. And the other worry people have is that only large companies can create GMOs. Yes, that is certainly true, but there are some examples where universities have created these GMOs. Papaya, virus-resistant papaya is a good example of that. It was created in Cornell University. There is a very small company that has created this apple, non-browning apple, and the public institution Embrapa in Brazil has created these virus-resistant beans that are available in market in Brazil. So it is possible to have traditional transgenic technology done by universities or other public institutions. And other myth that is out there is that the farmers, only large farmers benefit, and that is not true. Economic analysis has been done, and people have found that for $1 of investment in GM seeds, the farmers in developing countries are getting $4 back. So they're benefiting from this. And with that, I will just introduce few people. Leanne has been with me for a long time. Dave has been with me for also a very long time. These are the people who have done some of the work that I've done, but over the 25 years, I've had a lot of people working on these technologies, and a lot of work has been funded by these agencies, and I've had a lot of collaborators from all over the world. And with that, I'll stop and be happy to answer any questions that you may have. Thank you.
Thank you, Professor Arthur, for very inspiring information regarding the gene. I would like to read first the comment through chat room. Then, are you ready to be there, Professor Arthur?
Yes, I'm available.
Okay.
Okay, I'm going to have to stop sharing here. Yes.
Okay. The first question is, as methods in your The first will be, as method in your presentations, agrobacterium is a promoting technology for generating crop resistance to common abiotic stress. Would you explain the prospects of pairing agrobacterial technology with the recent advance related technology, such as tissue culture, etc., please?
Okay, I'm not fully understanding this question, but I'll answer it anyway. So the tissue culture process is totally different. So that's just the cell culture. So agrobacterium is a technology, or we use the agrobacterium technology to introduce a transgene into a cell, and then that cell has to go and develop into a whole plant or fertile plant, and that's all the tissue culture process. But if the question is whether more advanced technologies, agrobacterium is certainly used in conjunction with genome editing technology that I talked about, for example, using the CRISPR-Cas9 technology. So you can introduce the CRISPR-Cas9 components into a plant. The agrobacterium will introduce Cas9 and the guide RNA. Once the target gene has been mutated, you can use traditional plant breeding methods to then eliminate the CRISPR-Cas9 components. So that's, you can still use it. Now, the protoplast system, which is the alternative technology, and the gene gun is another technology that can introduce these things. Both these technologies can, they do have one advantage in that you can introduce the Cas9 enzyme and the RNA, so, and you can carry out those mutations, and there is no integration of transgenes, so you can call your plant non-transgenic. You can label them as non-transgenic, so that's one advantage. However, as I mentioned, that even with the agrobacterium system, you can eliminate the CRISPR components using the traditional breeding, and then at least in the US, that plant from which the CRISPR components have been eliminated will be considered non-transgenic.
Thank you for the time being. That's feedback response to Muhammad Akil. And then the second comment will become from Pak Nyoman Widiarta. His question is, numerous newly released superior varieties did not adopted by farmer. What is your advice to the breeders to make their varieties being adopted?
Okay, if the question is related to transgenic varieties that are superior, I'm assuming that is the question. Then I would say that again, the limitation is not the farmers. The limitation is the regulatory agencies. So, for example, let's take one example, BT cotton. Okay, so BT cotton was created here in America, and some of the other countries in South America, Brazil and Argentina, adopted it very easily. But a country like India, for example, they thought that this technology is needed in India, but the government was extremely slow. And the farmers wanted this technology because they knew that this technology can help them. So, and finally, government relented. It's a long story, but farmers, finally the government had to listen to the farmers, and they accepted this technology. And India is growing 80 to 90% of the cotton being grown in India is BT cotton. So that's one example. So the farmers, I would say, are not resistant to this. Now, obviously, sometimes the cost comes into place. Obviously, if the technology is created by big companies, they want to make some profit out of that. So the price of those seeds is going to be higher. But as I've shown you with my example, and I've given you, 2 or 3 other examples, where people have created these things in public institutions. So, for example, the trade that we have created, the gossipol trade, our university is very willing to give that out to other countries on humanitarian basis at no cost.
Yes, that's for Pak Nyoman. And then we still have 2 comments from the chatting room. The third will be, what can advance to the fact that the wheat grows faster than transgenic plants? Or what will be the consequences since wheat grows faster than transgenic plants?
Okay, all right. I think this person in the audience probably did not fully understand the slides that I was showing. The weeds will grow faster than transgenic plants only when you are fertilizing with phosphate, PO4. But if you fertilize with PO3 or phosphite, the transgenic plant that has the PTXD gene can easily outcompete the weeds. So that's, so I think, because the time is short, maybe I wasn't able to go into too much detail showing those slides. So the slides that you saw on the left side were, those 2 trays were fertilized with PO4, normal phosphate, which is what most farmers use. But the trays on the other side were fertilized with phosphite. And when you use phosphite, you can suppress the weed growth. I hope it answers the question.
Thank you. That's the feedback to Mr. Cyril Njume. And then the last comment from the chatting room is from Bapak Muhammad Judawi. Transgenic plants are types of plants that are produced from genetic technology processes through gene transformations. With the existence of transgenic plants, food needs can be met from a small number of plant seeds. In addition to having a positive impact, because transgenic plants are the results of technology. Of course, they still have a negative impact on humans and environment. The question is, what's the biggest negative impact of transgenic technology? Or the conclusion is, what's the negative impact of the technology in transgenic plants? Please.
That's a very good question. So I will be the last person to say that any kind of genetic transformation is good. That's not the case. So this is such a powerful technology that you can even produce, I'll give you one example, you can even produce human growth hormone in a maize plant, for example, okay? Now that to me is a very bad use of technology, and it's not been done, but technically that is possible. So there are many things you can do, but those are the kind of things that are possible, but should never be done. Okay, so that's one example. The only other thing that I can think of that may have some negative impact is this. So one would be, say, Roundup resistance gene, which is a herbicide. Roundup is a herbicide. And so when farmers were growing a lot of crops, especially here in the US and some probably in Brazil and Argentina also, a lot of crops they were growing had this gene, Roundup resistance gene, and so they were using Roundup very liberally. And so some of the weeds have now developed resistance to Roundup. If the Roundup resistance gene was not there, this transgenic technology was not available, they probably will be using Roundup, maybe some other herbicides, one or the other. So the development of resistance may not have been this fast. Eventually, they will develop resistance no matter what you do. It's not because of the transgenic technology people were using, but it may have hastened the development of resistance. So that's one example that I can think of. But one thing you can be sure of, that the government agencies are extremely stringent. They look at each and every product that you create very, very, very carefully before they approve it. So take our example. So we had done the science in 2006 for this gossip oil project. So it took us from 2006 to 2019 before we got the approval. So we had to do so much work to satisfy the government agencies that what we have created is safe and it does not harm the environment or the people or the animals that will eat the product.
Thank you. All audiences, I would like to say that we still have 2 minutes more time, but because of the preparations, I need only one comment from the floor if there is. Yes, raise your hand. Okay. You may comment directly, please. Can you come in front? One question, Prof. Rator, please. One comment.
Thank you very much for this time. It's very interesting to us explained about genetic engineering of the plants or transgenic. I'm a breeder, but I'm working for the conventional system. So I would give you the first question. I know that transgenic, you are working in the small room only, in laboratory. How could you confidence that after you make selections in the small size, small rooms, pot experiment after you bring it to the field? Because the field is very, very complicated system. So if you find the genetic material, for instance, resistant in transgenic system in laboratory, it's the same character after you bring to the field. How could you explain it? The second question about in Indians, you are talking about mainly the sorghum and cotton. How could be control after you variety you distribute it to the farmers? How could you control, for instance, the increase of the seeds, for instance? For instance, maize, like me, I'm working to the maize. It's very important control to the farmers. We are the right for the inbreeding system. The farmers, it could be increased her seeds only. But what about the transgenic variety? How could be controlled after you distribute to the farmers in India? Thanks very much.
Is it cooperatory? It's clear, but let me repeat it.
Yeah, I don't fully understand the first question. What he asked, I guess, we do the initial work, obviously we do in the laboratory, and we do the work under greenhouse conditions. It's, as you probably noticed from the examples that I gave you, and it's not a full list. I only showed about 2 traits in sorghum and maybe 5 in cotton. And as you probably saw from my talk, there was only 1 trait that we have taken out to the field. What that shows you is that we are being very careful about what traits we take to the field. Okay. Sometimes we do this kind of work. Again, we are in the university, and a lot of our job is to train students and the postdocs and so on. So they carry out this kind of work. They're learning the technology, which hopefully someday they can use. When they get their jobs and everything. So, but we are very, very careful. The government does not give us permission even to grow 2 plants outside in the field. You have to get a permit every year, and you have to tell them exactly what you're doing, how big is your field, and surrounding the field, there is no other, say if I use cotton, there should not be any cotton grown within a certain distance of my field. Okay, so there are a lot of precautions we're taking. The government does not allow you to grow these things, just anything you can grow. So I hope that answers your question. The second one, again, I didn't fully understand. I guess your question was about India. The seeds that Indian farmers finally, the first batch of seeds that the Indian farmers were able to grow were originally created in Monsanto here in the US, and the farmers wanted those seeds. So they were demanding those seeds, and because they knew the benefits of that. And again, but that all has to be done in the right way, because even with the BT cotton, now there is a buildup of resistance in some of the insects. So I don't know if that answers your question, or whether that was the right question or not. I don't know. I'm not clear.
Thank you. I hope that it will be clear to Pak Yasin. So the last comment from Pak Yasin is actually the transgenic working from the small but deep in a room, and then we'll go to a large area. And then the question is, how far the consistency from the laboratory to the field? But the answer is both. Professor Arator said that it's safe, since we must have a permissions from the government before releasing any transgenic plants. Thank you to everybody. Thank you to Professor Arator and to all the participants that joining this international conference through line and offline also. We would like to meet in the next future event. And now let's give a big applause to the presenter. Thank you, MC, and then I give time again to you.
Thank you, Prof. Yunus Musa from Hasanuddin University. Distinguished speakers, participants, ladies and gentlemen, we have finished the second session. We will now have lunch break and Zuhur prayer for an hour.
We would like to inform you that the next session will be started at 1 p.m. in this room. Thank you.
Corn is one of the major and strategic crops commodities in Indonesia. The Indonesian government through the Ministry of Agriculture will continue to increase corn production to meet national demand. One of the Ministry of Agriculture's strategic programs for the 2015 to 2019 period is food self-sufficiency.
Kepada peserta sudah bisa makan siang
One of
di
the
depan.
technological component that accelerate corn productivity
Kemudian
is through the
kembali
introduction of new high-yielding variety
pada
by
pukul
adopting
1.
molecular-assisted selection technology.
Break time sampai dengan pukul 1.
Indonesian Agency for Agricultural Research and Development has released over 50 high-yielding hybrid variety, one of
Break
which
time
is NASA
sampai dengan
29.
jam
This new variety
13.00
has a high
waktu
yield
Indonesia
of
Tengah.
approximately 13 ton
The
per
surface
hectare,
o'clock.
double ear potential, good resistant to pests and diseases, adaptive to the environment, and highly preferred by farmers.
Offline presentation start.
Prior to release, NASA 29 has gone through multi-environmental testing and involving global experts from CIMMYT, Handong Global University of South Korea, and Shandong Academy Agricultural Science.
14 o'clock.
Dissemination of candidate variety were performed at various national and international events, such as World Food Day, PANOS, National Seeds Expo, and variety display held by IAARD. NASA 29 was officially launched by the President of the Republic of Indonesia, Joko Widodo, at World Food Day, on November 29, 2016, in Boyolali, Central Java.
Satu batang tanaman jagung bisa buahnya ada 2. Ini memang baru pertama ini. Nih, jadi hal-hal seperti ini menjadi kunci nantinya di dalam kita swasembada. Kuncinya di benih, udah. Dengan hektar yang sama, tapi hasilnya bisa.
Attention please.
After launched, this new high-yielding variety was introduced
We
to the
will
stakeholders
continue
and policymakers
this
through
conference
technological
at
display, exhibition.
1 p.m. and offline presentation start at 2 p.m. 14 p.m. Thank you.
IAARD is also responsible for monitoring and evaluation of the seed production to guarantee the quality of the seed produced by the private companies. Seed quality
Please
is one of the
enjoy
key factors that determine the
food
crop
and
production
drink
outcome. Therefore,
in the
the
room.
availability and the quality of the seed are needed to be considered. IAARD conducts quality control mechanisms in seed production through the application of quality management systems according to national and international standards and seed laboratory testing.
Kalau dulu Pak beli katakan yang namanya bibit jagung tuh susah bantuannya Pak. Karena masyarakat kami ini katakanlah yang selama ini ingin mengembangkan bibit jagung tapi bibitnya harus beli kan gitu. Alhamdulillah dibantu oleh pemerintah dengan bibit jagung NASA 29 ini mudah-mudahan berkembang.
Kalau jagung sudah hampir 2 tahun. Sementara ini kami jadi penangkarnya. Benihnya 5-20 uri. Mimpi saya memang kebutuhan jagung nasional akan terpenuhi dengan jagung anak bangsa. Saya yakin itu.
Nah, kalau jagung NASA 29 ini berkembang kan 1 hektar sampai 6 ton Pak. Bahkan sampai 12 ton. Nah kali 2.000 berarti kan sudah 24 juta. Nah, jadi petani kami itu tuh terbantu kan gitu, senang kan gitu. Mungkin bisa mencapai yang dikatakan kesejahteraan masyarakat.
Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations.
Traditional dance from South Sulawesi.
Dengan mengucapkan Bismillahirrahmanirrahim, the International Conference on Sustainable Cereal and Crops Production System in the Tropics, saya nyatakan dibuka dengan resmi.
Ladies and gentlemen, in the next future, we believe that the agricultural development will have so many challenges.
Selamat siang, nama saya Gabrielo, saya dari Madagaskar dan saya salah satu partisipan yang mempresentasi hasil penelitian selama International Conference yang berada di Makassar dari 23 sampai 25. Jadi kalau saya ditanya bagaimana testimoni saya dengan conference ini, pertama kali saya lihat bahwa levelnya konferensinya sudah bagus. Tematik-tematik yang di-share oleh presenter-presenternya bagus juga. Jadi saya kira sangat penting konferensi seperti ini buat anak-anak di Indonesia, buat yang masih muda di Indonesia, buat yang sudah high level di Indonesia untuk membuat Indonesia biar berkembang. Dan yang saya sangat suka juga adalah interaksi sama teman-teman, sharing tentang sesuatu di Sulawesi. Saya kira sangat-sangat baru buat saya.
Ini adalah suatu kegiatan yang sangat bagus dan strategis, terutama untuk para peneliti. Tadi kita sudah mendengarkan pemaparan yang sangat menarik dari pemateri atau narasumber kita dari luar negeri. Bagaimana ke depan dengan adanya kegiatan ini kita mampu meningkatkan
kapasitas sumber daya manusia peneliti kita. Kemudian kita bisa mendapatkan teknologi-teknologi baru, terutama tadi dijelaskan bagaimana double halfway yang bisa masukkan kegiatan varietas baru. Kemudian peluang kerjasama ke depan dengan adanya kegiatan ini, kita saling berinteraksi dengan negara-negara lain sehingga kita dapat bekerja sama dengan baik yang manfaatnya sangat besar karena ada pertukaran pengetahuan, pertukaran teknologi, dan kita juga bisa saling mengakses basis manfaat yang mereka miliki maupun yang kita miliki. Dengan demikian kita maju bersama untuk mendapatkan hasil yang terbaik. Yang terbaik yaitu kualitasnya tinggi, kemudian murah, ramah lingkungan, dan tentu untuk yang terakhir adalah kesejahteraan petani.
International Conference on Cereal ini sangat penting, strategis, dan sangat bermanfaat, terutama pada saat kita harus mendukung program nasional ketahanan pangan. Jadi para peneliti di beberapa negara hadir ke sini, ini sharing pengetahuan, sharing ide terhadap riset masa depan yang berbasis genom yang sangat diperlukan untuk negeri kita, negeri tetangga, dan juga seluruh negeri yang diwakili oleh para peserta. Oleh karena itu, pertemuan seperti ini sangat kita hargai upaya dari Balai Penelitian Sedaria dalam rangka mendukung ketahanan pangan Indonesia dan juga negara-negara sahabat.
corn is one of the major and strategic crops commodities in Indonesia. The Indonesian government through the Ministry of Agriculture will continue to increase corn production to meet national demand. One of the Ministry of Agriculture's strategic programs for the 2015 to 2019 period is food self-sufficiency. One of the technological components that accelerate corn productivity is through the introduction of new high-yielding variety by adopting molecular-assisted selection technology. Indonesian Agency for Agricultural Research and Development has released over 50 high-yielding hybrid variety, one of which is NASA 29. This new variety has a high yield of approximately 13 ton per hectare, double ear potential, good resistant to pests and diseases, adaptive to the environment, and highly preferred by farmers. Prior to release, NASA 29 has gone through multi-environmental testing and involving global experts from CIMMYT, Handong Global University of South Korea, and Shandong Academy Agricultural Science. Dissemination of candidate variety were performed at various national and international events, such as World Food Day, PANOS, National Seeds Expo, and variety display held by IAARD. NASA 29 was officially launched by the President of the Republic of Indonesia, Joko Widodo, at World Food Day on November 29, 2016, in Boyolali, Central Java.
Satu batang tanaman jagung bisa buahnya ada 2. Ini memang baru pertama ini. Nih, jadi hal-hal seperti ini menjadi kunci nantinya di dalam kita swasembada. Kuncinya di benih, udah. Dengan hektar yang sama, tapi hasilnya bisa double.
After launched, this new high-yielding variety was introduced to the stakeholders and policymakers through technological display, exhibition, and field day. IAARD will supply the parent seed and distribute it to the private companies for seed increase or F1 seed. IAARD is also responsible for monitoring and evaluation of the seed production to guarantee the quality of the seed produced by the private companies. Seed quality is one of the key factors that determine the crop production outcome. Therefore, the availability and the quality of the seed are needed to be considered. IAARD conducts quality control mechanisms in seed production through the application of quality management systems according to national and international standards and seed laboratory testing.
Kalau dulu Pak, boleh katakan yang namanya bibit jagung tuh susah bantuannya Pak. Karena masyarakat kami ini katakanlah yang selama ini ingin mengembangkan bibit jagung tapi bibitnya harus beli kan gitu. Alhamdulillah dibantu oleh pemerintah dengan bibit jagung NASA 29 ini mudah-mudahan berkembang.
Kalau jagung sudah hampir 2 tahun. Sementara ini kami jadi penangkarnya. Benihnya 5-20 uri. Mimpi saya memang kebutuhan jagung nasional akan terpenuhi dengan jagung anak bangsa. Saya yakin itu.
Nah, kalau jagung NASA 29 ini berkembang kan 1 hektar sampai 6 ton Pak. Bahkan sampai 12 ton. Nah kali 2.000 berarti kan sudah 24 juta. Nah, jadi petani kami itu terbantu kan gitu, senang kan gitu. Mungkin bisa mencapai yang dikatakan kesejahteraan masyarakat.
and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations.
Traditional dance from South Sulawesi.
Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations. Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations. Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations.
corn is one of the major and strategic crops commodities in Indonesia. The Indonesian government through the Ministry of Agriculture will continue to increase corn production to meet national demand. One of the Ministry of Agriculture's strategic programs for the 2015 to 2019 period is food self-sufficiency. One of the technological component that accelerate corn productivity is through the introduction of new high-yielding variety by adopting molecular-assisted selection technology. Indonesian Agency for Agricultural Research and Development has released over 50 high-yielding hybrid variety, one of which is NASA 29. This new variety has a high yield of approximately 13 ton per hectare, double ear potential, good resistant to pests and diseases, adaptive to the environment, and highly preferred by farmers. Prior to release, NASA 29 has gone through multi-environmental testing and involving global experts from CIMMIT, Handong Global University of South Korea, and Shandong Academy Agricultural Science. Dissemination of candidate variety were performed at various national and international events such as World Food Day, PANOS, National Seeds Expo, and variety display held by IAARD. NASA 29 was officially launched by the President of the Republic of Indonesia, Joko Widodo, at World Food Day on November 29, 2016, in Boyolali, Central Java.
Satu batang tanaman jagung bisa buahnya ada 2. Ini memang baru pertama ini. Nih, jadi hal-hal seperti ini menjadi kunci nantinya di dalam kita swasembada. Kuncinya di benih, udah. Dengan hektar yang sama, tapi hasilnya bisa double.
After launched, this new high-yielding variety was introduced to the stakeholders and policymakers through technological display, exhibition, and field day. IAARD will supply the parent seed and distribute it to the private companies for seed increase or F1 seed. IAARD is also responsible for monitoring and evaluation of the seed production to guarantee the quality of the seed produced by the private companies. Seed quality is one of the key factors that determine the crop production outcome. Therefore, the availability and the quality of the seed are needed to be considered. IAARD conducts quality control mechanisms in seed production through the application of quality management systems according to national and international standards and seed laboratory testing.
Kalau dulu Pak, boleh katakan yang namanya bibit jagung tuh susah bantuannya Pak. Karena masyarakat kami ini katakanlah yang selama ini ingin mengembangkan bibit jagung tapi bibitnya harus beli kan gitu. Alhamdulillah dibantu oleh pemerintah dengan bibit jagung NASA 29 ini mudah-mudahan berkembang.
Kalau jagung sudah hampir 2 tahun. Sementara ini kami jadi penangkarnya. Benihnya 5-20 uri. Mimpi saya memang kebutuhan jagung nasional akan terpenuhi dengan
pengumuman kepada co-host masing-masing ruangan.
Nah, kalau jagung NASA
Bapak Andi Iwan, Bapak Fitra, Ahmad Ali, dan Mas Oki Dwi Prayitno diharapkan untuk berkumpul.
Sudah 24 juta. Nah, jadi petani kami itu terbantu kan gitu, tenang kan gitu. Mungkin bisa mencapai yang dikatakan kesejahteraan masyarakat.
Tenang bro, tenang. Tenang co-host mengambil ruangan. Bola sobat satu. Bola sobat satu kan Bu, satu. Iya, terus bola sobat satu, bola satu.
Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food.
Yang ini aja dulu Pak, yang ada aja ruangan.
Agriculture plays important role in the national economic development to achieve food security.
Kalau yang udah ada nanti dia biar langsung masuk gitu. Berarti bola sobat satu, bola yang kedua.
Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in
Bola sobat satu.
1974.
Bola sobat satu itu untuk apa? Mbak, tentukan Mbak bola sobat
Until
satu.
today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations. Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations.
Goyang dua. Bola sobat satu. Mas Odep. Mas Odep di ruang VVIP HPT. Hama penyakit tanaman. Ya, nanti namanya pest and disease ya.
An important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations. Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations.
Keluar nih. Silakan masuk.
Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations. and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through innovation. Food had and has always been an important issue in the world. All countries try their best to fulfill the need for food. Agriculture plays important role in the national economic development to achieve food security. Indonesian Agency for Agricultural Research and Development, or known as IARD, Ministry of Agriculture, was established in 1974. Until today, IARD has strategic role to improve agriculture efficiency and competitiveness, as well as contributing significant development on agricultural technology through its innovations.
Halo, Dr. Naik. Dimohon untuk semua peserta bisa kembali ke ruangan karena acara mau dimulai kembali. Dimohon untuk semua peserta diharap kembali ke ruangan karena acara segera dimulai kembali. Terima kasih.
Kepada Bapak dan Ibu para peserta konferensi yang masih berada di luar, dimohon untuk segera memasuki ruangan karena sesi berikutnya akan segera kita mulai.
Sekali lagi kami informasikan kepada Bapak Ibu partisipan peserta konferensi internasional pada hari ini agar dapat bergabung kembali ke dalam ruangan. Sebentar lagi sesi berikutnya akan dimulai.
Dimohon kepada panitia yang berada di luar untuk mengarahkan kepada para peserta memasuki ruangan karena acara akan segera kita mulai. Bapak dan Ibu para partisipan yang kami hormati, insya Allah kita akan masih mendengarkan satu materi lagi, kemudian dilanjutkan dengan diskusi secara berkelompok. Acara akan berlangsung sampai besok hari ya. Jadi kepada Bapak dan Ibu yang masih berada di luar, agar acara kita berjalan sesuai dengan waktunya, dimohon kepada Bapak dan Ibu untuk segera memasuki ruangan karena acara akan segera kita mulai.
Baik, kami informasikan kepada Bapak Ibu dan juga seluruh partisipan yang masih berada di luar ruangan agar dapat bergabung dan mengisi tempat yang telah disediakan. Sebentar lagi acara atau sesi berikutnya akan kita mulai. May we have your attention please? The next session is about to begin. We kindly ask you to resume your seat. Thank you.
Distinguished speakers, participants, ladies and gentlemen. For the next session, we are honored to have the third speaker, Dr. Yu Shin Nai, a Professor of Insect Pathogen, National Chungsin University, Taiwan.
Presenting his paper, Construction of an Entomopathogenic Fungal Library for Controlling Fall Armyworms, Podoptera frugiperda, and a case study of integrated microbial control of pests. And the chairman of this session is Dr. I. Ketut Suar Adnyana from Dwi Jendra University.
Okay, thank you for MC and for the opportunity given to me, and I hope after having lunch, all participants have more energy to attend this conference. The Honorable Director of ISRI and Director General of IEERD, Rector, and all participant. I am Ketut Suar Adnyana from Dwi Jendra University Denpasar, Bali. I am pleased to meet you and to see you here, and welcome to all of you to this conference. Ladies and gentlemen, we have a speaker from, I mean that our speaker today is Dr. Shin Nai from National Chung Hing University, Thailand. Before the presentation will begin, I would like to greet Mr. Yu Shin Nai. Hello?
Yeah.
Mr. Yu Shin Nai?
Yeah. Hi. Hello.
Okay, thank you. Before the presentation will be begin, I would like to introduce the CV of Dr. Yu Shin Nai from National Chung Hing University. The title of his presentation is Construction of an Entomopathogenic Fungal for Controlling Fall Armyworm and a case study of integrated microbial control of pests. Dr. Yu Shin Nai, PhD, the work history, I would like to introduce as one work history, and from February 2020 till now, as an assistant professor in the Department of Entomology, National Chung Hing University, Taiwan. And the background of education, Dr. Shin, Dr. Shin took his BS degree in 2000 till 2004 in Department of Entomology, National Chung Hing University, Taiwan, and finished his master degree in Department of Entomology, National Taiwan University, Taiwan, and received PhD degree in Department of Entomology, National Taiwan University, Taiwan. And honor received September 2014 Best Paper Award, the 15th Conference of Korean Society of Sericultural Science. Okay, I think it's enough for CV from Dr. Yu Shin. Okay, now we come to presentation session. I would like to welcome to Dr. Yusin to present his presentation, and the floor is yours.
So I have to share my PowerPoint, right? It's me. So everyone can see my PowerPoint? Is everyone see the PowerPoint?
No, not yet.
Okay. It's normal one or with some speech note? It's normal one, right? Okay. It's normal one. Yeah, I think now it's okay, right? So may I start?
Sure.
Okay. Okay, good afternoon, everyone. Thank you for the moderators introduce me. And my name is Na Yuxin from the Department of Entomology at the National Zhongxin University. So first of all, I'm very honored to be one of the invited speakers in the second international conference on sustainable cereal and crop production system in the tropic. Okay, today I would like to share the topic about the construction of an entomopathogenic fungal library for controlling fall armyworm. And also in the second part of my presentations, I would like to share some cases for the concept we called integrated microbial control of pests. Okay. So before we start into talk today, I would like to briefly introduce myself again, although the moderator already introduced me very detailed. I'm a graduate from the Department of Entomology at National Zhongxin University, and in 2004, and I got my master's degree and a PhD degree in the Department of Entomology at National Taiwan University in 5 years. But trust me, got a PhD degree within short times is not a very good idea, actually. My major research is in insect bacterioviruses. Therefore, my PhD thesis was focusing on the genomic sequencing and analysis of one insect bacterioviruses, lemnichus and multiple respiratory virus, which was found in Taiwan. Okay, so my current research topics are focusing on the insect pathology and the insect pathogen genomics, microbial control, and the honeybee pathology to protect the honeybee from the disease. Yes. Okay. Here's my work list. I know it's a quite long one. Just briefly, after I got a PhD degree, I started to do a postdoc in National Taiwan University and in the genomic research centers at academic clinical until 2013. And then I found a research assistant professor position in Chongbuk National University at Southern Korea for 1 year. After 1 year, I got a chance to become the project assistant professor in National Ilan University for 3 years until 2018. Finally, I come back to my mother university, National Zhongxin University, and found the assistant professor positions in 2020. Although it took almost 11 more years to find a formal job for me, actually, I think every job position or every work period are very meaningful for me because until now, I still have some connections with this the postdoc positions in my current research. Okay, when I come back to National Zhongxin University, I starting to set up my lab, and I think the most important things is to set up research directions for short-term and long-term research in my career life. Here is my lab website. On this website, I listed 4 different research directions, including the first one is very important one called diversity of insect pathogens. In these directions, in these research directions, I have to find more and more highly potential insect pathogens for the further applications or further studies. So I call the first directions is diversity of insect pathogens. The second one is applications of insect pathogens. After I found some highly potential microbiome of insect, and then I can try to establish them as a microbiome control agents for further applications. It's called application of insect pathogen. The third one is deepness of insect pathologies, which means after I found these insect pathogens, and I can use this material to do the research of the interaction between the host and the insect of these insect pathogens. And the last one is called dynamic of insect pathogens. This kind of research about the beneficial insect disease. So if you are interested on what we are doing on the insect pathology, you can scan the QR code in the right down side, and then you can get into our lab website, and then you can check what we are doing now. Okay. So based on the 4 research directions, I listed the current goals in my lab. The first one is safeguard of agriculture by discovering and developing new microbial and genetic-based strategy for pest control. It's very important for the future. And the second goal is study the insect pathogen and the host interaction by omics technique. For example, the genomics, transcriptomics, and the proteomics to more comprehensive to understand this kind of interactions. So for the first goal, the first step for me is to find the new and potential microbial control agents or insect pathogen which can develop into the microbial control agents in the futures. And so if I got such kind of insect pathogens, then I can get the ticket to study more or modified by the genetic-based strategy for best pest control. And in this slide, this is a very simple classification of the most famous microbial control agents, including Bacillus thuringiensis for bacterial entomopathogens and Barreria bassiana, Metarhizium, and Nisophis for entomopathogenic fungi. Of course, also include the Chinese style of microbial control agents called nuclear polyhedrovirus (NPV). And also I list one Microsporadia microbial control agents, and Antonospora locusti is a kind of pathogen for the controlling the locust. Okay, so I listed several kind of commercial products of insect pathogens, but the question is very important. The question is very important for me. Of course, you can buy the commercial product in any store, or there is how if we can find some insect pathogens that maybe much better than the commercial one. Yeah, that's our chance. So I started to my research generally to find a new and better insect pathogen. So therefore, in the following slide, I would like to briefly share one of my current research projects. Okay, sorry, it's a little bit trouble for my internet. So I'm back. Can you hear me? Yes.
Yes, Dr. Nai, we can hear you.
Hey.
Thank you.
Okay, therefore, in the following slide, I would like to briefly share my current research projects on the entomopathogen fungi, and I believe this project has potential to reach my goal for a safeguard of agriculture in the future. This project is establishing and development of insect microbial resources for controlling pests. So actually, this project was starting when I worked as a research assistant professor in Chungbu National University in South Korea at 2014. And in that time, I worked really very hard with Professor Jeju Kim, and we are trying to collect the soil sample all around the South Korea and try to isolate the entomopathogen fungi from this soil sample. As you can see, the left picture shows the red spot indicate where we go and to collect the soil sample. So it's almost around the South Korea. And then that right picture shows how we work in the field. So we love barbecue. So after I come back to Taiwan, I started start to entomopathogen fungi isolation and the selection work from 2018 until now. Okay, this picture shows how did we collect the soil sample and isolate the entomopathogen fungi. Actually, we collect the soil sample in different mountain areas in Taiwan, and then put the mealworm larvae onto the surface of the soil sample, and wait for 2 weeks. After 2 weeks, we can find the necrosis on the surface of the mealworm, and this fungal infected sample will be taken out for fungal isolation and selection. And also, we will record the detailed information for the mountain area which we collect. However, the process of fungal isolation and selection of very heavy loading work for me. If I can I doing myself, it will take a long time. Therefore, when I come back to National Chongqing University, I started to set up a SOP for animal pathogen fungi isolation and selections, and starting to recruit more and more students, and starting to training them to do the routine job for me. And here is the SOP. As you can see, and this SOP includes three major part. The first part is the first one is the isolation of entomopathogen fungi from the soil sample, and the second one is pathogenicity screening of this isolated entomopathogen fungi. And that's the last step is molecular identification of this entomopathogen fungi based on the ITA's regions and to the genus level classification. Okay, and this is one of my students. He was doing the molecular identification of entomopathogen fungi. After we got the DNA sequence of ITAs, we will perform the phylogenetic analysis to make sure the classification positions of this entomopathogen fungi isolate. Okay, in summary, until now, I have checked yesterday. Until now, we have isolated 263 fungal isolates from the soil. But among these fungal isolates, there are only 100 fungi showed insect killing activity, which would mean this 100 fungi may be animal pathogen fungi. And most of them will belong to the genus of Metarhizium, Breviaria, and Fusarium, based on the ITA sequence. And also we upload our data on the iCloud and to save this information. Okay, to test the insect killing activity of this animal pathogen fungi isolate, we perform a pathogenicity screening against several kind of crop pests, including the mouse-tail, aphid, cabbage-flea beetles, tobacco cutworm. And this picture shows the mycosis of the animal pathogen fungi infected insect. And very soon, we have got a success on the establishment the animal pathogen fungi library. And we suddenly note there have another purpose of the animal pathogen fungal library construction. That is through the establishment and development of insect microbial results, we can quickly respond to the invasion pest species such as fall armyworm. Yes. As you know, the fall armyworm is a notorious and a very serious pest species all around the world. Unfortunately, fall armyworm invaded into Taiwan in 2019. As you can see the pictures, the red area indicates the distribution of fall armyworm adult, which was surveyed by the sex pheromone trap. The population of fall armyworm break out throughout Taiwan in 2020 until now, especially in the southern east of Taiwan, as you can see here. Okay. How terrible of fall armyworm? I think I don't need to very detailed to describe how terrible of fall armyworm, but it does really have a wide host range, especially they feed on mice and rice, and they also cause a lot of economic damage and controlling costs every year. The most important point is the problem of insecticide resistance. It makes the farmer very helpless to control the fall armyworm larva. Therefore, for our researcher, we have to think about an alternative strategy to control this kind of pests. And the animal pathogen fungi within the genera Breviaria and Metarhizium provide an alternative choice for biological control against the fall armyworm, as we know. So we started to test the pathogenicity of this selected entomopathogenic fungi against the 4-amino larva. Here is the result of pathogenicity screening against the 4-amino worm. There are total 28 entomopathogenic fungi isolated, and most of them are showed a high variance, as you can see the heat map here. Most of the entomopathogenic fungi we selected show high variance against the 4-amino larva. And the right side shows the mitosis of 4-amino worm after fungal infection. Finally, we select 13 entomopathogenic fungi isolate that show fast killing and higher variance to 4-amino larva for our further studies. And this is the phylogenetic analysis of these 13 entomopathogenic fungi isolate. Based on the result, there are total 2 species in the genera of Bavaria and 3 species in the genera of Metarhizium. So even though entomopathogenic fungi within this genus, they are composed of many kind of species. So until now, I think all things maybe things be done very well, but I ask one question to myself. There are so many research groups isolate and select a lot of entomopathogenic fungi, not only in Taiwan, I think all around the world, so many research groups have do the same job in their lab. So many entomopathogenic fungi isolate were selected and trying to control the pest. But however, why do these entomopathogenic fungi still difficult to apply to the agriculture system? Is it a problem of mass productions, or because of the environmental stress, or because of the virulence decrease after several pathogens? I think all of these questions are very important for us for the next step for applications of entomopathogenic fungi. To solve this problem, I try to symbolize these kind of problems. I start to think about how if we can set up the stress selection system in lab conditions and ranking the entomopathogenic fungi isolate with higher stress tolerance. Therefore, I choose 2 parameters, including conidia productions and the thermal tolerance, to roughly establish the ranking system of entomopathogenic fungi in our lab. Okay, this is the result of conidia productions of 13 entomopathogenic fungi isolate. As we can see, in general, Bavaria species usually have a higher conidia production than that of Metarhizium species. Among Bavaria species, NCHU 157 showed highest conidia production value, and NCHU 9 showed highest conidia production value among the Metarhizium species, as you can see here. And for the thermal tolerance, this is the result of thermal tolerance of 13 entomopathogenic fungi. We found that the Metarhizium species show higher thermal tolerance than those of the Bavaria species. And among these isolate, NCHU 124 show highest thermal tolerance within the Metarhizium species, and NCHU 153 showed highest thermal tolerance within the Bavaria species, as you can see here. Okay, so I therefore combine the data of conidia productions and thermal tolerance to generate roughly generate a formula of effective conidia spore number, and to combine these 2 sets of the data. Through this formula, we can combine more and more data together to rank the top 1 or top 2 entomopathogenic isolate with the higher stress tolerance. Based on this result, as you can see, Metarhizium NCHU 124 and Bavaria NCHU 157 were selected for the further bioassay. So this is bioassay of the two selected entomopathogenic fungi against the 4-amino larva. And the upside pictures shows how we do the bioassay. And the results show that these two selected entomopathogenic fungi isolates show a high virulence against the 4-amino larva and also show a dose-dependent effect to the 4-amino larvae. However, I definitely sure about that every researcher like me to do the entomopathogenic fungi research should be asked the same question in the final stage for the commercialize. Okay, the question is, for entomopathogenic fungi, the time for killing pest is still slower than the chemical. It's incompatible, right? Usually, as we know, microbial control agent usually have a slower insect killing activity than that of chemical. But I started to think about one idea. This is a concept. So here I want to propose this concept that's called integrated microbial control of pests, we call IMCP. This concept could be performed by the following three ways. The first strategy is combination of microbial and other adjuvant to enhance the insecticidal activity of this combination formula. The second strategy is combination of different microbiomes to present the synergy effect of this formula. The last one is combination of microbiome and some epigenetic regulators, for example, the double-strand RNA, to suppress or to roll down the past insect performance and increase the insecticidal activity of this formula. Okay, in the following session, I will show you the three different cases for the integrated microbial control of pests. So the first one is a combination of microbial and other adjuvant to enhance the insecticidal activity. Before I started the second part, do you think about the concept can work very well or not? Please keep the answer in your mind, and I will give you the answer later. Okay. As we mentioned before, there are two selected promising entomopathogenic fungi isolates show the high virulence against the 4-amino larvae. And we performed the bioassay as well. And we found that those two selected entomopathogenic fungi strain show the dose dependence effect. But in the bioassay data, we can find that lowest concentrate of 10 to 6 conidia per milliliters of these entomopathogenic fungi show very low insect killing activity. As you can see, the green line here, the mortality is very low here with low concentrate of entomopathogenic fungi, and which lower than 50 mortalities, even though we cannot calculate the LT50 here. But after we adding some adjuvant to the low concentrate 10 to 6 conidia per milliliters of the entomopathogenic fungi, so the new combinations formula can dramatically enhance the insecticidal activity of the entomopathogenic fungi. As you can see here, the upside pictures shows the original data without any adjuvant, but in the down red part pictures, you can see after we adding the adjuvant, the combination formula showed a higher mortality, the increased mortality dramatically. Okay. Moreover, after we adding the adjuvant to the lower concentrate of the selected promising entomopathogenic fungi, this formulation can dramatically decrease the time for killing the 4-amino larva. As we can see, the table shows the original data that we can see that the 10 to 6 conidia per milliliter cannot calculate any LT50 data. However, the increase of the variance with 2 to 5 days after we adding the different concentrate of the adjuvant. Therefore, it's really work. Right? These combinations reveal a great efficiency for controlling farmer larvae. Besides, we also found the mycosis of entomopathogen fungi formulations could still found in the embedded farmer larvae. So let's come back to the questions from the beginnings. Is the concept of integrated microbial control of pests concept work? Yes, the question is yes, and it can work very well. And as our data show that this kind of combination is very great and useful for the sustainable agriculture application in the future. Okay, the last two cases are still ongoing work in my lab, but I can still show the some possibility to the INCP concept, and hope in the future we will have a large progress on this project. The second case is a combination of different microbial fields to present the synergy effect. But before that, I want to talk a short story. In this part, I would like to share an RNA virus identified from the transcriptome data set of fungal-infected beanbags. In the beginning, our goals were trying to better understand when the beanbags infected with entomopathogen fungi Bassiana, what is the response of the host insect, and what is the gene expressed in the entomopathogen fungi Bassiana. So we're starting to do the experiment and collect RNA sample and subject this RNA sample to the RNA-seq and to do the transcriptomic analysis. And the right picture shows our experiment design. Okay, so if you don't understand or you have any idea about RNA-seq, here is some simplified experiment flowchart for you. I think I can skip this part, right? And if you have any further question about this technique, please email me and I will give you a detailed answer about what is metagenomics or what is transcriptomics analysis. And after RNA-seq and the data analysis, we found that several transcripts with a highly expressed level. And the most interesting thing is that this highly expressed transcript neither belong to the beanbags nor belong to the entomopathogen fungi Bassiana. Besides, they are belong to genomic RNA of RNA viruses. So therefore, we chose the highest fortunate counting, which was C43419G1I1 counting for further analysis and provisionally land it as RIPV1 based on the host species. So we then started to assemble the genome of RIPV1 and predict the open region fragment of this virus. As you can see, the total length of this RIPV1 is around 10 kilobase, and here is the protein coding regions, including helicase, protease, or RNA-dependent RNA polymerase, and some structural protein regions located in the 3' end. And also we conduct the 3' and 5' race to complete the whole genome sequence of RIPV1. So here is the phylogenetic analysis based on the RNA-dependent RNA polymerase domain. According to the result, RIPV1 is a brand new and novel RNA viruses which found in the beanbags, and it belongs to the on the side in the RNA virus group. It means we found a novel RNA virus through the transcriptomic data. Okay, to further demonstrate the existence of RIPV1 in the bean box, we started to collect the bean box sample from Korea and subject to the viral purification and the electronic microscope observation by negative staining. As the data show here, we found a really viral particle here with a icosahedral structure. Finally, we identified two RNA viruses from the set of our transcriptome data. But as you can see that the previous slide showed the table, there are still many data need to be identified. They are all belong to the RNA virus. But the most important thing is in this slide. During our experiment, we also found that the replication of RIP1 will increase dramatically in the bean bags which infected with entomopathogenic fungi. After we sprayed in the fungal conidia suspensions 2 to 6 days, the RIP1 replication was dramatically increased, higher than the control. So therefore, this is a very fantastic case for me to give us a hint that insect RNA viruses may be potentially work as a synergy pathogens for insect pathogen-based microbial control, integrated microbial control microbial control. Maybe in the future we can use this concept to find more RNA virus and combine with bacteria or some entomopathogenic fungi microbial control agent to generate one new commercialized formulation or commercialized product for our further applications. Okay, the last case is combination of microbial and epigenetic regulator to suppress the pest performance and increase the insecticidal activity. But as we know that the insect express many genes to against the environmental stress. For example, the expression of immune-related genes can help the insect host against microbial infections, and the expression of detoxification genes may contribute to the resistance of the insect. Therefore, the concept of the the concept of the epigenetic regulators is use double-stranded RNA to knock down or to suppress the insect host genes to suppress the insect performance and make them to more susceptible to the fungal or bacterial or viral infection, even though more susceptible for the pesticide. Okay, this project is cooperated with Professor Chen Yihong at the Department of Plant Pathology in our university. Now we have chosen 4 GMBP genes of 4-amine which responds to the fungal and the bacterial infections. In the future, we will also test the efficiency of the double-strand RNA and the combination of microbial, including infect with the entomopathogenic fungi and the bacteria, and compromise with the double-strand RNA to see what we can get in the future. Okay, in the summary, okay, we in the beginning we have constructed an entomopathogenic fungi library in Taiwan, and after the pathogenicity screening, we have select around 100 entomopathogenic fungi isolate, and the entomopathogenic fungal library can help us to quickly respond to the emerging pest species such as 4-amine as our talk today. Besides, in today's talk, we also present the stress selection system, and this system can help us to select the entomopathogenic fungal isolates with higher stress tolerance in the environment. To improve the efficiency of the biocontrol agents, we propose a concept of the integrated biocontrol of pests. And in this concept, we can combine the microbiome with different microbiome control agents, with different adjuvant, with different epigenetics regulators. So today we present 3 different case studies, and I think the first one is very work for the further applications. The work of adjuvant plus showed a great enhanced efficiency to the microbiome for killing the foreign larvae. So the other 2 strategies, although are still ongoing job in our lab, but I think in the future it will be very useful for the microbiome control agents. And the final slide is a take-home message for you. And recently, a concept of pest biome was proposed. In this concept, a healthy gut microbiota present the higher diversities in the gut of insect, but somehow there are some environmental factors can lead to the disease status to the insect, in which the gut microbiota showed a low diversity. So if we can use this concept and we can find that the integrated microbiome control strategy may be a force to make the insect microbiota to become a pathobiome status. So in the future, we can generate a lot more and more ideas to induce the pathobiome status for the pest insect species. And then we can enhance our microbiome control agents in the future. Okay, I would like to thank, this is, I think, in the final slide. I would like to thank all the lab members in our lab, and including the research assistant Zhang Rujun and the master student in the master programs, Yao Jia, Chen Lu, Yixuan, Zihao, and Jin Chen, and some undergraduate students, Nian Tong and Fang Min in our lab. This job are all done by the students. And also, I would like to thank my supervisor, Professor Wang, although he already passed before, and the other cooperator groups, including the National University Group and the Korean Group and the GRC in Academic Syndicate groups. And also thanks the most to support me, the funding to do the research, and the RDA in South Korea and some research institutes in Taichung. Okay, thank you for your listening and attentions. This is my presentation.
Give applause. Thank you very much, Dr. Yusin Nai, for giving us such informative and interesting presentation. We come now the answer and question session, but before we begin this session, okay, I would like to say to the committee to show the list of questions on the screen. Yeah, because my handphone cannot connect to the link. Can you show, please?
Ya, sudah Mas.
Okay, this is the list question. Maybe Dr. Yusin Nai, can you read the list of question on your screen?
Yeah, I can. Yeah. So for the questions, I can answer one by one, right?
Okay.
You have a question to come about the...
Okay. Can you answered for the first question from Muhammad Akil. Please,
Yep.
Dr.
So the
Yusenai.
question is, yeah, okay, use the tiny grinders for set pasture. Use we do our experiment by the Cornelia spore, so which is a only a Cornelia spore. So to make the Cornelia suspensions. So for the tiny granular, we didn't use the tiny gran for the animal pathogen for controlling the armyworm. Okay, so is it possible to integrate to a drum bass spray? Yeah, of course. So, but before use a drum bass spraying technique, we have to test. Yeah, this is a very good idea. But in Taiwan, the area for agriculture crops growth is maybe not as bigger as worthy to use the drum. But maybe in the other countries, they have a bigger area for the agriculture crops. It is worthy to develop such kind of technique by using drum to spray the Cornelia or some formulations to the crop system. Yeah, it's a very good idea. Okay. So second question is
Next to the second session, Dr.
Yeah, second
Yusenai.
section. Yeah, biocontrol agency in agriculture is often Christian that yeah. The same thing happened in how can you become an biocontrol agency in agriculture so my comment for the tropic area for use the micro biocontrol. In Taiwan, we also, between the subtropic and the tropic area. So we've also faced the same situations. Usually we isolate the microbial control, insect pathogens from lab. And then if we want to apply this kind of material to the agriculture system, they are usually useless, or they cannot have a very good efficiency for the controlling of an insect. So my idea, I also present in the presentations. My idea is to use the stress selection system to select in the laboratory. You can use higher temperature to culture or to select the microbial control agents from the environment, and then you can get the thermal tolerance strain. And another idea is you can make a genetic exchange by some technique, for example, the electrophoresis, to make the genetic exchange and to get the high thermal tolerance or high UV tolerance or high stress tolerance strain, and then you can use this strain to do the field test. So this is my idea about the situations. Okay. And the other one, the other idea is maybe we can use the formulation. Some folks, I think some material can protect the microorganisms, protect them from the UV and some high temperatures in the environment. So the formulation is also very important for us. And in my presentation, I showed the adjuvant. Maybe some adjuvant can also play the role of protect the microorganisms as well. Okay, so this is my answer for the second questions. Yeah, the third question is there any possibility of the pest that will be resistant to the animal pathogen fungi? Actually, let's back to the mechanism of how animal pathogen fungi get into or infection to the insect. As a Cornelia spore, as a starting stage for the infection process, after the Cornelia spore attack to the insect cuticle, there were germinations and become the epithelium stage, and then they will try to disrupt the structure of the insect cuticle, right? So it is very hard to generate resistance by the entomoplasmogen fungus. But as I know, BT already have some resistant insect. Yeah, the insect already have some resistance to the Bacillus aeruginosus. But for entomoplasmogen fungus, until now, I still didn't see any reference. If you have any idea, you can email me and we can discuss about that. But until now, as I know, it's very hard to have a resistance of entomoplasmogen fungus. But the host range is very interesting topic because we found that maybe the same strain, the same species with different strain, they can still have a different host range. Yeah, it's a very interesting research topic. Maybe, yeah, we can do this research topic in the future. Okay. So the third question is, so I need your opinion. What is the difference in the principle in the degraded pest management? Yeah, in use of control threshold to insecticide with pathogen fungi. It's a very good question, and for the concept I proposed today is just only based on the microbial control agent and use some non-chemical agents to enhance or to make a synergy effect. So the base of the concept is only based on the microorganism. This is what I think. Okay. So do I have any time to answer more questions?
I think it's enough,
okay.
Dr. Yusenai, because we are limited by time.
Okay. Yeah.
Thank you for your.
I can use the question and give you the answers and the email to the conference, right?
Yeah. Okay. Okay. Thank you, Dr. Yusenai,
okay.
For such informative and interesting presentation, and we give applause here to the Dr. Yusenai. Thank you. I don't make a summary for this session because Dr. Yusenai have explained and do summary for his presentation. Okay, thank you.
Thank you. Thank you. Bye.
For all participants, we finish with the all invited speakers for today. Today we're gonna continue to the online with the parallel section. So we divide into 5 rooms. In here gonna be a offline room, and then there are another 4 online room. Wait. So for the other room gonna be the ruangan nggak tahu kan. Oh iya. Untuk for room A, B, C, and D, you need to connect to the each link. So here we attach the meeting ID and a passcode. So later you can join the each room. Depends on what topic you want to listen. So for room A is a breeding and biotechnology. Room B is crop production system. Room C is pest and disease management, and room D is crop production system. Yeah. Thank you. Yes. Offline gonna be here. Yes. So for all the presenters who already join and already go through the room A gonna be offline gonna be here, and also this room gonna be divided into two room A and offline and room D, and then room B gonna be in the bola. Iya, tapi lupa namanya bola apa, Bu? Iya, bola soba 1 for room B, and then room C gonna be in boyang, and then room D gonna be here, VIP. No, VIP. C gonna be in the VIP room. Please sign into each meeting code for all the presenters who gonna be the oral presenter. Thank you so much. We're gonna start at 2 p.m. So it's now. Thank you.
Good afternoon, everyone. So, for you who will present it offline, you will be staying here in this room, and I will be chairing the session. So, I have here the list of how many is this? 8 titles of presentation that will be delivered in this room. The first presenter will be the team of Bapak Amran Muiz, and the title is the field testing of synthetic insecticides on fall armyworm in corn plant, and then followed by a presentation by the team of Bapak Agung Gunawan, entitled Vegetative Growth Response of Upland Rice to Actinomycetes, Azospirillum, and Azobacter. May we request the presence of the first presenter, Professor Amran Muiz from the Indonesian Cereals Research Institute. Our IT friend, could you please help to lower the volume of the audio so that we can start with the presentation? So while we were waiting for Pak Amran to load the presentation into the computer, I'd like to inform you that for all the oral presenter will be given separate certificate of appreciation, only for the oral presenter. While for the rest of you who attend this meeting, you will be given a certificate of attendance. You may get the certificate by filling up the registration form, which the link is available at the entrance desk. You may check it later. Pak Amran, are you ready to present now? Okay, sir. It's supposed to hear. Our IT team, please, IT team, show the presentation of Pak Amran's team. Belum dinyala. If not, then you may use my mic. Oh, kok di sini? Kok di sebelah? Can we move it?
Ntar panitia punya pointer gak? Pointer pen?
So, Pak Amran, you will have 7 minutes for a presentation.
Okay.
And then later on, we will do the question and answers at the end of after all the presenters' presentation.
Okay. Thank you, Dr. Nuning. Good afternoon, ladies and gentlemen. I would like to ask Pak Sahrir and Ibu Nina, please stay here in this room because this is our presentation. The title of our the title of our paper is Field Testing of Synthetic Insecticide on Fall Armyworm. Unlike Dr. Nai just now, that he applied entomopathogen, but still, as we know that entomopathogen, the work of entomopathogen took a long time for effectiveness. This is, next please, Abdal. As we know that some of insect pests in corn, like Ostrina furnicalis, Helicobacter parmigiana, Mithymna separata, Peregrino, Sesame inferens, Sporoptera litura, Sporoptera examptera. Last year, Spodoptera exampta attacked quite wide of corn plantation in central Sulawesi. And Peregrinus, we saw that insect 2 years ago in South Kalimantan. And We have Spodoptera frugiviridae here, or fall armyworm, and Plosia calcita. Next, please. Fall armyworm, one of the most destructive pests in the world, including corn. There are more than 80 crops as a host of fall armyworm. This pest first reported in Lampung and West Sumatra in March 2019. From 2019 to 2021, the pest was found in all crop production area in Indonesia, from Sumatra to Papua. The most destructive state is larvae, attacking corn crops from the start of plant growth. It can cause yield loss up to 100%. It depends on the age of crop were attacked. Next. This is the symptom of fall armyworm in the field. Next, please. Our trial is located in Bajeng. Bajeng Agricultural Technology Research and Assessment from March to August 2020. Atau kita sebut dengan IP2TP. Variety use HG21, HG21. Ini haji jagungnya Pak Takdir, hibrida jagung. Then the treatment, 3 replicates. The treatment including 4 synthetic insecticide, namely emamectin benzoate, spinosauteram, carbofuran, chlorantraniliprol, and 1 botanical pesticide, citronella, and a control without pesticide. We apply 2 times in vegetative stage and 2 times in generative stage. Next. The plot size is 5 times 5 meter, planting space. Our next, please. The scale of observation, we use Davis scale from 0 to 9. Next, please. This is the result. Our observation on 2 weeks after planting, we didn't see the result yet because we did not apply yet pesticide. The next 2 weeks after applying insecticide, we see the result. The spinosauteram, there are 3 kind of insecticide. There are 3 insecticide has low incidence of fall armyworm. Next, please. This is the 6 WAP, 8 weeks after planting, and so on. Next, please. After 10 weeks after planting, still these 3 insecticides have a very good action in controlling fall armyworm. Next, please. Then the yield we have spinosauteram have a higher yield compared to others. Spinosauteram insecticide compared to control, very much different. Next, please. Then we have conclusion. The best control method for armyworm application of synthetic insecticide is, we emphasize here synthetic insecticide. Insecticide that are effective in expressing, WF, not WF, we have highly work from home. It's supposed to be fall armyworm there. Spinosauteram, Ini ngerjainnya jam 10 malam ini. Ivermectin and so on. Spinoteram insecticide can reduce yield loss due to fall armyworm. Next, please. Udah? Okay, thank you.
Join me to give a round of applause for Pak Amran and the team. Thank you, Pak Amran. Very good work. I hope you will stay in this room because we will have discussion later on.
Of course.
So now we are proceed to the second presenter. The team of Bapak Agung Gunawan will presenting vegetative growth response of upland rice to, okay, this is difficult for me, actinomycetes, azospirillum, and azotobacter. Ibu will be presented. Okay, you may use. Amran, don't bring home the mic, Pak Amran. Ibu, please, this is the mic. Oh, okay, yeah, you may use that also as well.
Okay. Oh, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. This is time for me to present my paper. This is the collaborative research by the team, and I am the second author. I'm Yusmi Nahala. Today I will present my paper about vegetative growth response of upland rice to actinomyces, azospirillum, and azotobacter. Next. Okay. Upland rice is a rice grown on dry soil rather than flooded rice paddies. Nearly 100 million people now depend on upland rice as their daily staple food. Upland rice is grown in rain-fed fields, prepared and seeded when dry, much like wheat and maize. The ecosystem of the upland rice is extremely diverse, including fields that are level, gently rolling on steppe at altitude up to 2,000 meters and with rainfall ranging from 1,000 to 4,500 millimeter annually. Although the rice technology of the 1980s and 70s focused on irrigated rice, rice is the upland rice not forgotten. The challenge facing scientists is to supply high-yielding growth-promoting factor from rhizosphere bacteria adapted to the poor soil of the uplands. In this case, we use azospirillum, actinomycetes, and azotobacter that isolate from rhizosphere of Mimba. And the mechanism to fixing nitrogen by plants, it's accommodated by bacteria that produce, that get fixing nitrogen from the atmosphere. Next. Now, the method in this research is consists of the test the suitability of nitrogen bacteria with upland rice seeds. In this case, we use actinomycetes, azospirillum, and azotobacter bacteria are inoculated to upland rice seed to test the ability to associate with upland rice in vitro. Seed priming was performed against 50 upland rice seeds soaked for 24 hours in pure isolate suspension, actinomycetes, azospirillum, and azotobacter with concentration of 10 for 8 to 10 for 9 cc CFU per milliliter. Next. And the second is the test the ability of nitrogen fixing bacteria into plant on upland rice growth. The bacteria, the nitrogen fixing bacteria suspended by mixing natrium chloride 0.9% with several bacteria colonies taken using sterile use and incubated for 48 hours to obtain bacteria with concentration of 10.4A to 10.9 CFU per milliliter. Next. Next, please. And the third is how to processing of planting media. The media that we use is soil, about 2 kg per polybag of plants, and then the soil is sterilized by using autoclave. And the fourth is upland rice plant maintenance. This about upland rice plant are watered 5 times in a week and just to the level of soil depth. The parameter is measured about 3 weeks, 5 weeks, and 7 weeks after planting. Next, please. And the fifth is measurement of vegetative parameters of upland rice grain. This is about a high plant, a number of rice tiller, and biomass. This is about dry wet roots and the top of rice plant. And the sixth is the measurement of nitrogen content of upland rice plant. Next, please. This is also a measure about the soil physical and chemical properties analysis and measurement of plant growth that the content of the parameter about the number of tiller, plant hay, plant and root dry weight, and etc. Next, please. The result of the research show that the application of actinomycetes, azospirillum, azotobacter on the length of seed of the upland rice show that the application of azotobacter gives the very high length radicule seed, but it's not different significantly. The result also appear in the upland rice plant hay. Yes, the application of azotobacter also gives the highest result, but it's not significantly different with another bacteria. Okay, next. This is also about the rice tiller. After 7 weeks after planting, the measurement show that application of azotobacter give the highest number of tillering, but it's also not different significantly with another bacteria. This is also the do at the heavy dry of top part of the plant. This is also give a result like this. Okay, next. The result of heavy dry roots of upland rice plant shows like the result also like another parameter. The appearance of the plant that inoculate by azotobacter give the very high result, but it also not significantly different with another bacteria. Okay, next.
2 more minutes.
Yeah. Okay, I think all of the parameter shows the appearance like one another. Next, please. Yeah, this is also, now at the table 10 we saw that the quality of the soil before and after planting show that there are increasing quality of the soil. That's about the soil texture from clay to clay, pH, that's about from low to medium. That's a very advantage for the soil. And total nitrogen, it showed that from low to high, available V, available phosphor, it showed from low to high, and the available potassium is from low to high. Okay, next. And the conclusion show that all of the parameters show that the application of azotobacter give very high result, but it's not different significantly with another treatment. I think that's all. Thank
you very much. Join me to give a round of applause for the presenter. Thank you, Ibu. So before we move to the next presenter, I would like to ask the help of Pak Ayub or somebody to put it down the podium so that everyone can see the screen without any disturbance. Diturunin aja, di sini aja, taruh di sini. Ya. Okay. Thank you. Okay, so ladies and gentlemen, we are now proceeding to the next presenter. So the next presentation entitled The Distribution Model of ASI. I suppose this is anthesis silking interval, I don't know, under anthocyanin corn. Will be delivered by Bapak Tarmizi AS or his team. May I request the presence of the team of Bapak Tarmizi? Oh, Pak Yasin yang akan presentasi. You will be the one to deliver the speech, Pak. Okay. Asrot ready with the presentation? So after this presentation, we will continue with the presentation by the team of Ibu Risma Eva Cahyanti, determination of total phenolic. I hope he or she will be already ready here. Okay. Pak Yasin, time is yours.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. For this afternoon, I would be trying to talking about my one of experiment in the Islamic University of Makassar. The title is The Effect of ASI under Anthocyanin corn. So we are 5 researchers. The first is Tarmizi, Yasin, me, and Rahman. It's the teacher in OIM, Hanafi and Jumiarti. This is the Tarmizi is the experiment of his thesis for finishing study in S1 program. Okay. ASI, the main subject, I will be talking about ASI, distribution of the regressions model exponential of ASI. ASI is anthesis silking interval. Anthesis silking interval. We define of different periods between silk. Silk is going out from the stem as female is silk to pollen seed male in the up of the plant and in days. I give you an instance, if silk coming out is 55 days, and female 50 days, so ASI is 5 days. So the difference between silk period to the anti-silking intervals to pollen seed, which defines the ASI. ASI is very, very important for the selection of the maize, eventually inbreed once plants or population. Is very important because if the ASI is not synchronized, exactly we cannot find the maximum of the yield. It's the not selected for the plant population or inbreed. So it's very, very important. It's the main character for selection of the core to generate of new variety for founded of high yield, because we cannot find yield if the plant, the ASI is not synchronized between female period to the pollen seed. If more than 7 days, this is the experience. If you find the plant hybrid, the ASI is more than 7 days, it's delayed. Don't continue to the next generations, because exactly we cannot find the maximum of the yield. It's the introduction of the ASI, anti-silking interval. Yes? Okay, the objective of this experiment to found the exponential regression model of ASI. Jadi ASI we put in the ordinate from 0, 1, 2, 3, 4, 5, and 6, 7. The model could be used for corn selection to find out of the best yield. So this is the regression mathematics for found the exponential between ASI to the characters of the ears of the crops. So we are not observed to the vegetative stage of the plant. We are concentrated to the ear or crops. Okay. So this is the experiment we conducted in farm of Agricultural Faculty, Islamic University of Makassar, since October to December 2019. So purple population names ikan di Umuwan. We are planting in the garden 70 to 20 centimeters, one plant for one hill. ASI as treatment, we are defined from 0, 1, 2, 3, 4, 5, 6, and 7 days. ASI was started process to compute between ear to close by silk back as female and tassel back. So this is the treatment. We are a conventional system by manual. So after we find the 2 centimeter and direct to cross pollinate, it's the name 0 treatment. When we check the silk around 2 centimeter and take pollen and pollinate, it's defined to 1 days. Until around 8 to 10 centimeters, the length of the silk, and we take pollen and to pollinate, it's defined 7 days. So this is the treatment work conducted by the pollinate manual system, not by wind or by insect. Yeah. So the tassel was covered in around 20% and take for pollinate. If they are flying insect around the tassel, we have to be carefully started for the treatment from 1, 2 until 7 days. It's the take for initial of the treatment. After data we conducted, we observed the analysis we compute by model of regression exponential. Y is alpha E rank beta X. And we transformation into basic logarithm, ln A. This is the reference from Smith, Dr. Beck. If you analyze of the ASI of the maize, you don't make multi-regression or simple regression, but you have to, his conclusion, we have to approach by exponential regression model. So this is the recommendation for MCMIT we applied here to want to find which the best of ASI under anthocyanin corn. Okay. The result and discussions. The first, we applied the simple regression correlation between variables. This is the variable we're computing from the ear aspect only. S1 is the ear length, panjang tongkol. X2, diameter of the ears. X3, number of seed in rows, jumlah biji di dalam satu baris.
3 more minutes, Pak Yasin.
Okay, X5, number of seed. You can find the coefficient of the beta, which the good is X4. X4 is number of seed per row. It's very, very important to increase the yield of the anthocyanin corn. And it's not important for the selection, and X5 minus 4.1. X5 is number of seed per year. Okay? This is the objective of the experiment. We want to find the exponential ASI to the grain. This is the parameters for the grain weight, gram per plant. The conclusions, let's grab is the red color. We find Y is 160.7. E rank minus 0.3. This is the good exponential ASI for anthocyanin corn. What we could be conclude the disease model, we could find the maximal of the yield if we find ASI less than 0 days. We still find 160 gram per plant. This is the per plant. Less than 2 days. You can see ASI when the yield, we cannot find yield in the year. And the grain weight, we cannot find after 6 days. Okay? This is the conclusion to the result of exponential ASI for anthocyanin corns. Y is 160.7, E rank minus 0.3. And the termination coefficient is 51%. So high yield could be found and the value of ASI is 0 in this experiment. But in the field we can find the formation is less than 2 days. Some days crosses of mile to pollen. From that variable, we could find 161 gram per plant. So we conclude also, if you find the plant ASI is more than 7 days, don't take it. No yield could be found. This is the conclusion of my experiment. Okay? You can find that this is the treatment. So when the silk could be closed by the plastic. When in the right picture, it is the treatment 7 days of ASI, around 10 to 12, the length of the silk. We are defined the 7 days. Okay. So the manual system. When after we are cover of the silk, the next is the pollen seed. So we cross like this. You can find that when the pollen is going out more than 20%, we close with the tassel bag. And we wait treatment by the check of the silk after covered by the plastic bag. Yeah.
Okay, thank you, Pak Yasin. Your time is up. Sorry.
System.
Your time is up, Pak Yasin. I have to apologize to interrupt you. Give a round of applause for Pak Yasin and the team. Thank you very much, Pak Yasin. I'm very happy to see you after your retirement, you are still eager to research, and that really give us an inspiration. Yes. Thank you, Pak Yasin. Okay, so now we are moving to the team of Ibu Risma Eva Cahyanti. We'll be delivering the title is quite long, but I will read it carefully. Determination of total phenolic content and antioxidant activity of a mixture of tomatoes. Okay, mixed fruit. From Malino, Goa Regency, South Sulawesi. Ibu Hala will present again. Oh, okay.
Thank you very much for the second chance. I'm Ismina Hala. I'm from Universitas Negeri Makassar. This is my team research, the next team research of Risma Eva Cahyanti and Andi Munisa. The title of this research is Determination of Total Phenolic Content and Antioxidant Activity of Fruit Mix from Malino, Goa Regency, South Sulawesi. Next, please. Okay, as introduction, the lifestyle is the healthy lifestyle is a lifestyle that pays attention to all aspects of health condition, ranging from food, drink, nutrition, consume, and daily behavior. For some people, living a healthy life is not easy, especially for those who have busy work. So they prefer to consume fast food and drinks. However, consuming fast food and drinks in excess can cause various diseases caused by an increase in free radicals. Free radicals are molecules fragments that contain one or more unpaired electrons in their orbitals. Free radicals are considered dangerous because they can be very reactive in gaining their electron pair. Due to its highly reactive nature and irregular movement, free radical can cause damage to various parts of living cells and can cause various dangerous degenerative disease. The human body can neutralize free radical in the amount is not excessive with an antioxidant defense mechanism. Antioxidants are compounds that can prevent and slow down the damage caused by free radical through the inhibition of oxidative mechanism. Increased production of free radicals due to stress factor, excessive consumption of fast food and drinks, ultraviolet radiation, and air and environmental pollution resulted in inadequate defense system, so additional antioxidants from outside the body were needed. And antioxidant outside the body can be obtained in synthetic and natural form. Antioxidant compounds that are proven to have high antioxidant potential and activity are found in fruits and vegetables that contain vitamin C, vitamin E, beta-carotene, and plant that contain phenolic compound. Okay, next, please. Okay, the problem of the research is what is the total phenolic content in the extract of tomatoes, purple passion fruit, Nimarkisa, and strawberries. And the second is what is the antioxidant activity of extract of tomato, purple passion fruit, and strawberries. And the third is what is the total phenolic content and antioxidant activity of a mixture of tomatoes, purple fruit, and strawberries. Next slide, please. Next, please. Okay, the method is how to prepare the sample fruit. The 3 tomato, purple passion fruit, and strawberries were washed and then dried, and each fruit was weighed as much as 5,500 grams. The wet fruit is mashed using a blender. This all of the fruits it's come from Malino, Goa Regency. For extraction sample extraction was carried out using the maceration method. Fruit sample that have been mashed in the form of juice are put into glass jar and soaked in 500 milliliter 96% ethanol. Next, please. Next, please. Okay. The photochemical analysis, this is about the alkaloid test, consists of Warner test, Mayer test, flavonoid test, terpenoid and steroid test, tannin test, saponin test. To determination of phenolic content of fruit extract, use 0.05 grams of each fruit extract was diluted in 5 milliliter of distilled water. The fruit solution mixture was taking 20 microliter, added 1.58 milliliter H2O and 100 microliter Folin culture reagent, added 300 microliter natrium carbonate, measurement of the absorbance of fruit extract. Next. The measurement of antioxidant activity using 77 microliter of fruit extract solution was put into test tube at 3 milliliter of DPPH solution, measurement of the absorbance of fruit extract. And the last is pH measurement. Samples of tomatoes, purple passion fruit, and strawberries before and after mixing were measured using pH method to determine the degree of acidity or pH of each fruit. And the result shows that the comparison of yield percentage of tomato, purple passion fruit, and strawberry extract before and after mixing, we can see that the mixing of fruit is not different significantly from tomato, purple passion fruit, and strawberries. Okay, next. From the phytochemical screening result of tomato, passion fruit, and strawberry extract, it's shown that the compound of the number of alkaloid, flavonoid, terpenoid, and steroid, tannin, and saponin. It looks like the all of the fruit that used in this research have a positive reaction. Only for steroid is no of them. Okay. Next, please. And this is the total phenolic fruit extract. The total phenolic fruit extract seems that the total phenolic is not different significantly from single fruit if we compare with the mixed fruit. Okay. Next, please. And this is about the antioxidant activity of fruit. It's we can see the treatment of tomato. This tomato. Previous. Okay. This from tomato, purple passion fruit, and strawberry. If use the single fruit, we can see that the antioxidant activity, it's not, it look like if you mix the fruit, it gives antioxidant activity is very high. We can see that the average of DPPH percent is about 88.32A. This is very high if you compare with the single fruit. But it's not like different, yes, not different, yeah, before, if we compare with the single fruit, okay? Next, please.
1 more minute.
Yeah. And this is the degree of acidity. We can see that if you mix the fruit, it's lower the pH. Next, please. Yeah, the conclusion is so that the total phenolic content of extract being equivalent of gallic acid, so that the highest total phenolic content was found in strawberry extract, which was 1.9, and then tomato fruit, and the last is purple passion fruit. The antioxidant activity of extract of tomato, purple passion, and strawberry showed the highest antioxidant activity was found in the extract of strawberry. And the total phenolic content is the highest we found in strawberry also. Okay, I think that's all. Thank you very much for your kind attention.
Thank you very much. Give them a round of applause for Ibu Hala and the team. So this result give me an idea that I should eat more strawberries rather than banana. Okay. If I want to look younger. Yeah. Okay, so we are now, So we are now we are now proceeding to the next presenter, the team of Ibu Nur Asiah Jainuddin will be presenting compatibility of biocontrol agent, formulas, and synthetic fungicides in controlling maize leaf blight on corn caused by bipolaris maize. Ibu Asiah, time is yours. Ini apa yang itu ya tadi? Ini kayaknya deh. Sorry.
Thank you, Chairman. Hi, everybody. Assalamualaikum warahmatullahi wabarakatuh. I'm so glad to be here because I can share a result of our paper, and we hope this research can accepted and utilized by many people, especially for farmers. And here's I'm gonna do it. I wanna show how compatibility of biological control agents, formulas, and synthetic fungicides in controlling maize leaf blight on corn. And that's our team, Nur Asiah Jainuddin, Amila Sebayang, Nurnina Nonci, and Amran Muiz, and we are Indonesian Cereals Research Institute researcher. And yes, let's get started. As research background, we know plant disease are still one of the limiting factors in increasing maize production, and actually the application biological control agents we call BCA in the future as a biopesticide to control pests and plant disease expected to decrease synthetic fungicide and also its impact. And besides that, several researchers show that formula biopesticide are suppress development of a major disease in plant. Next. And we know there is no single system can control pest and plant disease. It needs integrated disease control strategy with combination biological, physical, chemical control holistically. So this purpose of this study was to determine compatibility BCS and synthetic fungicide to severity as maize leaf blight. Next. We move to methodology. There are 5 types synthetic materials were used investigated. There are difenoconazole, propinep, fluopicolate, metalaxyl, and dimetamorph. And we can see dosage recommendation on the right column respectively. And then 3 types of synthetic active ingredients which have the highest of inhibitory effectiveness in laboratory tests were selected for field assessment. And inhibition effectiveness, synthetic active ingredients was calculated based on the formula diameter control minus diameter of treatment per diameter of control times 100. Next. And for field trial, the treatment were arranged using randomized block design with 2 factors. The first factor was biological control agents with 2 levels, T0 without BCA, T1 with BCA, and the second factor was synthetic fungicides with 4 levels, S0 without synthetic fungicide, S1 difenoconazole, S2 propinep, dan S3 fluopicolate, and each combination treatment was repeated 4 times. Next. Observation variables included the intensity of leaf blight 4 times, plant height 2 times, and crop yields, and severity of leaf blight scale based on Sharma 9083, and with 5 scale, and the scale of observation transformed to the following of percentage infection formula as seen on the screen. Next. We move to the result. For the first result from the laboratory test, we can see the effectiveness of synthetic fungicide inhibition on the growth of Miscelia diameter of Bimydis. We can see the inhibition efficiency at 3 days after isolation. All synthetic fungicide tested cause inhibition, which tended to be very significant in the growth of Bimydis, and fungicide active ingredient difenoconazole was found to be most effectively and compare the other treatment, and show their consistent at the 5 days after isolation, followed by propinep and fluopicolate. Next. Okay. There are several figures from the laboratory test. On the left figure, we can see Bimydis on toxic medium. Synthetic fungicide fluopicolate and propinep were observed profit equal inhibition of Bimydis, while BCAs suppress the growth of fungal by forming a clear zone around the fungal colonies, and while the treatment control, no pesticide, fungal growth normally. And the right figure, we can see compatibility testing between BCA and the active ingredient, and difenoconazole was compatible with BCA, characterized by absence a clear zone around the filter paper, and then other than that, fluopicolate was compatible too with the BCA, but propinep was less compatible with BCA. Next. And the next slide, we can see results from the field trial. This the percentage of severity Mydis leaf blight. We can see the application of biological control agent, BCA, combined with synthetic fungicide, had a significant effect to a rate of disease infection. We can see the treatment difenoconazole combined with BCA was found the lowest severity of the disease. Next. And the figure was visualization of interaction between factors. We can see the red line was BCA treatment and the blue line was without BCA treatment, and the disease severity of without BCA treatment or a blue line had significantly higher than red line or BCA treatment. Okay, next. The last result. The last result, we can see application of application of BCA and fungicide, pesticide, fungicide synthetic from the treatment of various types of pesticide on maize treated with bimidis. We can see treatment with single BCA or control was observed to be not significantly different from the BCA plus difenoconazole treatment, but significantly different from other treatment. Yeah. Next. Okay, at the end, I would conclude my presentation with the first one. There is a positive synergy between biological control agent and synthetic fungicide difenoconazole in controlling bimidis leaf blight on maize. And the second one, fungicide with AA difenoconazole was the most compatible with the lowest disease severity value, was 27%, while control treatment condition without BCAs was 72%. And that's, ladies and gentlemen, what I wanted to share, and thank you.
Thank you. Give him a round of applause for Ibu Nur Asia Jayonudin and the team. Next, may I call on the presence of Bapak Arif Muazzam or the team, who will deliver the observation of promising tungro-resistant superior rice lines in West Sulawesi. Bapak Arif, oh, ada di sini Bapak Arif. Okay. You may choose which one you like. Okay. Pak Arif, you have 7 minutes to present, please.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, please allow me to introduce myself. My name is Arif Muazzam. I collaborate with Mr. Mansur and Mrs. Sumarni Panikai. Next. Our research is about observation of promising tungro-resistant rice line in West Sulawesi. Next. Rice is one of the most important foods and a major food source of more than 1/3 of the world population. To meet food needs that are in line with population growth, the development of super rice varieties is very important. Superior parent material for breeding is necessary, so if we have good parent material, we will get lines more satisfied. Next. Rice production, for generally, there are so many problems. Irrigation, land, fertilizer, pest attacks, and destructive diseases, and so on. Tungro disease is one of the most important diseases of rice because it has the potential to cause quite high damage. Tungro virus disease caused by rice tungro bacilliform virus, ATBV, and rice tungro spherical virus, RTSV. To prevent a decrease in production due to tungro attack, it is necessary to assemble and develop the new superior tungro-resistant rice by utilizing genetic results for breeding of Tungro virus-resistant rice plant. Breeding activities to produce a new site-specific rice varieties resistant to Tungro quite require of suitable genetic material as cross-breeding material. Effort to establish or improve the characteristic of superior varieties need diversity based on phenotypic and genotypic character. Observation breeding material lines from screening needed to be done with checker varieties in endemic Tungro area. The purpose of this research was to determine the phenotypic character and yield of promising rice line Tungro resistant virus, which have truly good yield and resistant to Tungro. Next. We can see this is Tungro virus vector that cause Tungro virus in rice. Next. Material and methods. The research was conducted at Tungro virus research station, experimental garden and Peru district Kaluku village, Mamuju, West Sulawesi, in the second growing season of 2019 with 25 breeding material lines to see its resistance to Tungro virus and some of its agronomic properties. The crop was compiled in augmented design, consisting of 5 blocks. Each block there are 5 breeding material lines and 2 checker varieties, namely Ciherang and Inpari 36 Landrang. The time was adjusted to peak vector population. Each line was planted in plot size 1 × 5 meters, planting distance in 25 centimeters × 25 centimeters. Fertilizer used in 300 kilos per hectare. NPK Ponska 50 kilogram per hectare. Urea Ponska fertilizer and 1/3 of urea are given when the plant is 10 after planting, which 2/3 of urea is given at the age of 45 after planting or based on local farmer habits. Next. Observations were made on 10 sample plants that were randomly selected in each plot. The components were observed included the Tungro's incidence, plant height, flowering age, number of panicle, number of filled grain per panicle, number of empty grain per panicle, 1,000 grain weight, yield weight per hectare, clump shape, and dry field grain. Population of green leaf hopper. The observation of the population of green leaf hopper, name and imagus, were carried out using insect nets as much as 10 double swings per observation plot. Then the catch was put into the observation box, and then the population of green leaf hopper was calculated in name and imago phases. Observations were made 20 days after planting and 30 days after planting. Productive tillers are tillers that emerge before 7 weeks after sowing. The more tillers, it is hoped that the rice plant will have many productive tillers so that an increase in production can be achieved. To get the number of productive tillers, it is done by counting the number of tillers with panicle and seed containing them. Productive tillers were observed when the plant were 90 days after planting by counting the number of plants that produce panicle. Next.
3 more minutes.
Next. Result and discussion. The presence of Tungro attack in Mamuju on test land breeding material as well as check varieties show no difference There was no attack on the crops, although the study was conducted in endemic area. This is indicate that tungro occur when there are vector insect, inoculum source, infected with tungro virus, and rice that are susceptible to tungro. Even though the population of green leaf hopper is not always increasing, the percentage of attack of tungro disease. Mamuju condition as an endemic location, which is not supported by environmental or weather, can lead to endemic location shift, so there is no tungro attack. Next. From this table, we can see there is no difference promising tungro line with checker varieties because there is no tungro incidence. Next. Population density of green leaf hopper, the most we found in 4 weeks after planting 8 tail of green leaf hopper, and nympha, and for green leaf hopper, imagus in 4 weeks after planting. Next. Lines that are less favor green leaf hopper have the potential as a candidate for resistant varieties. Plants that have a certain morphology affect the level of preference of green leaf hopper to the plant. Next. Yield and yield component of rice promising resistant tungro line. For about 11 promising tungro line, rice plant, the yield much higher than checker material plant varieties. Next. This is 11 promising rice line that more higher in yield compare with varieties checker in variety 36 and Ciherang. Next. Our conclusion, the result show that in Mamuju, recorded all rice breeding line of tungro resistant test and check varieties with low vector population of neem and imago have no percentage of tungro attack. Lines that have no neem or green leaf hoppers imago, no tungro accident clumps, upright, compact clumps are not broken, and production is higher than checker varieties for about 5 promising line rice. While the line that have high production for about 11 promising rice tungro lines. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Pak Arif and the team. Round of applause for him and the team. And now we move to the last 2 presenters. First by Ibu Dr. Ramlah Arif and the team, entitled The Influence of Seed Priming on Vigor of Sorghum. Ibu Ramlah, you may choose which mic you would like to use. Okay, so you have 7 minutes, Bu Ramlah. And then later on we will have the team of Ibu Amelia Sebayang to present exploration of entomopathogenic fungi in Central Java. Okay.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Firstly, I would like to say thank to the moderator. Before I present my priming study on sorghum seed, let me introduce myself. My name is Ramlah Arif, And this is my team. We are from Indonesian Cereal Research Institute. Next. Oh, before, oh, not this. Okay, I will. This is, I have make editing a new one. Okay, no matter. I will, before, I just want to say, before I conduct this research, we have already published many research in line with this research. In the newer one, the research published research of priming has already in many publication. Why I choose sorghum in this research? Because sorghum has many advantages for food, feed, and industrial purposes. In Indonesia, sorghum we can find in East Nusa Tenggara, Southeast Sulawesi, and a little in Central Java. But you know, in East Nusa Tenggara, in Southeast Sulawesi, especially in these two areas, sorghum has already developed as a food, even only in small-scale industries. ICRI has already released many varieties of sorghum, but how we can have high productivity of sorghum crop, one of them we have to put a qualified planting material like high fiber seed. How we can find the high fiber seed? The farmers, most of farmers only store their seed for the next planting season. In the store, there are many factors affecting the seed quality. Once we store the seed, the quality will not increase. We have only maintain the seed quality, not to deteriorate rapid. Okay, one technique to increase viability and vigor of seed by we call as priming, or invigoration or conditioning. In this research, I use kalium nitrate salt solution to make the seed, to soften the seed. And the method called hydration, rehydration. Okay, the research aimed to evaluate influence of salt solution, in this case is kalium nitrate solution, as priming material to some combination characteristics of sorghum. Next. Okay, I use 3 kinds of sorghum, freshly harvested seeds, 12 months storage, and 24 months storage. And this is the treatments, control, and concentration, several concentration of kalium nitrate. Okay, observation on viability and vigor of the seeds. Next. The results. From the research, we found that kalium nitrate with 1.5% was the best treatment to increase seed vigor of sorghum. In terms of germination, next, germination rate. Next, seedling dry weight. Next, oh no, Feiger index and others. Other characteristic of seed, Feiger and viability. Okay. The conclusion of this research, we found that shorter seed period from 3 kind of seed, from freshly harvested seed, 12-month storage, and 24-month storage, that newly freshly harvested seed produce better seed. It's okay. And we found also that priming treatment with kalium nitrate 1.5% induce higher seed germination, about 5.1 to 16.3%. Germination rate 1.9 until 12.5%, and seedling dry weight 12 until 55.6%. So high seedling dry weight. What does it mean? It means that if seedling dry weight is high, percentage increase is high, that means that seedling Feiger index is also high. Okay, thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi. Thank you, Ibu Ramla. An applause for you. We apologize for some technical miscommunication about the presentation file, because we don't get the new file, you just get this file, so sorry for the inconvenience. Last but not least, I would like to invite Amelia Sebayang and the team to present to us the exploration of entomopathogenic fungi in Central Java, South and North Sulawesi, Central Java, Central Java, South Sulawesi, and North Sulawesi as potential biocontrol of corn earworm or Helicoverpa. Amelia, floor is yours.
Thank you so much. Good afternoon, everybody. Yes, hopefully we still have energy still because we start from the morning, and then today gonna be my last but not least, I hope. So, yes, presentation. Okay, let me introduce myself. My name is Amelia Sebayang. I am from Indonesian Cereal Research Institute. Today I'm gonna present my research. It's a collaboration research titled Exploration of Entomopathogenic Fungi as a Potential Biocontrol of Corn Earworm or Helicoverpa armigera Hubner. This research is a collaboration between me, Amelia Sebayang, and also Andi Tenrirawe and also Hisar Misram. Next, please. Thank you. Yes. So yes, like in the topic, I'm talking about the Helicoverpa armigera Hubner, or we can call it also Heliotis armigera. It's known as a common lepidopteran, as a polypagous insect, has 3 common names. Approved by Entomological Society, we can call them corn earworm, tomato fruit worm, and also bollworm. But in additional to being known as a sorghum headworm and also soybean padworm and also fetch worm. So this lepidopteran is a nocturnal insect. They have a 4 stage of life cycle. The life cycle is completed in 30 days. From egg into the adult, from eggs to larva, eggs is completely can hatch in 2 days, also single laid, and then going to the larva, and then pupa, and also the moth. The Adult moth is highly variable in color and wings markings. In addition to impact on yield, corn earworm injury the ears of the maize may provide a pathway of fungal colonization, leading the greater levels of the two major types of mycotoxin, fumonisins and also aflatoxins, which are major concern for human and animal. Next, please. So Helicoverpa armigera is annually estimate make a loss production is around $2 billion US, listed as an A2 quarantine pest by EPPO. And in Indonesia, this pest become important pest in corn because they make a cause yield loss until 51.9% until 53.4% in Sulawesi. So how to control this insect? There are many strategies to control this insect. One of it that we farmers usually use this strategies is using the pesticide. But this technique, of course, like the previous our invited keynote speaker also mentioned, is a damage our environment. So there are kind of strategies that we call IPM is insect pest management. There are collaboration with several strategies to control the insect. One of the strategies is we use the biological agents. What is biological agents that we can use is parasitoid, also pathogen. And in this research, I use pathogen to know how the pathogen can control this insect. Next, please. So what is the research problem? Biological control by using pathogen agents is an alternative control method that has been studied widely and considerably more effective and environmentally friendly. But less people believe with this. And also, biological control agents rarely eradicate the target organism. Yes, rarely eradicate the target organism, but reduce symptoms to an acceptable level and maintain the balance between the pathogen and target organism. So we are going to more sustainable to control the pest. So what is the research aim? Is this study aims to explore entomopathogen that attack Helicoverpa armigera, which will later be formulated and projected as candidates for biopesticides controlling Helicoverpa armigera. Next. So the research methodology, this research was taken in 3 areas. That was South Sulawesi, North Sulawesi, and also East Java. And then we also do in the laboratorium. We do insect rearing, pathogen isolation, morphology characterization in Israel pest and disease laboratorium. And then we do 3 main things to do. Here is surface and field collection, and then insect rearing and pathogen isolation, and then the third we do the morphological identification, that was macro and micro morphological. Next. So yes, what we do here, first we do insect rearing. We feed the insect and then we do routine cleaning. Why we need to do routine cleaning? We are afraid that the insect that we rear is affected by another organism, not the one that we want to understand. And then for the pathogen, so when we get the pathogen from the insect, we clean it with 1% sodium hypochlorite for 3 minutes, and then wash them with the distilled water. And then after that, we crush and dissolve them in 10 milliliter distilled water. The insect solution then rub on the larva food so that the larvae are infected. So this one is to multiply the pathogen. Next, please. So for the morphological identification, we do the initial propagation with PDA media, and then we do microscopic structures were observed with a light microscope with magnification 400 times, and then drip with lactopanol cotton blue to see more clear of the isolates. Next, please. And then for the result, we found that in Jawa Timur or East Java, in 3 locations, there was in Malang and 2 in Malang and 1 in Probolinggo, we found the name of the pathogen is Rhizopus, and then in South Sulawesi, we do in Bajeng, we found Beoveria and Rhizopus, and then in Malino we found Rhizopus and Aspergillus flavus, and then in North Sulawesi, we do in Mandoinding and Rurukan, we found Glycocladium and Rhizopus sp. Next. So here what we found, after we got the insect that we know that they are infected by the entomopathogen, then we do the to understand, to know what is the pathogen that inside. And then we found that there was Aspergillus and Beoveria in Bajeng 1 and Bajeng 2 also. We found Fusarium, and also in Malino we found the Aspergillus. So next. And also here we see the microscopic of the pathogen. After we found the macroscopic, and then we do the microscopic under the microscope. Here what we found, the shape of each pathogen that we found. Next. So as one as the pest control agents, entomopathogenic fungi have been enlightened for their ability to suppress pest population and crop damage. Abundant research of this pest control agent has been published, such as strains of Metarhizium and Beoveria to control eucalyptus, and also several research that have been conducted to understand how the entomopathogen can affect the work of the pest. Next. So what we conclude here is this research is a best of study. So it's just exploration of entomopathogen that we can find in the field. So what we can find here is there are several of entomopathogen that affect the pest actually. So we found Beoveria, we found Aspergillus, we found Glycocladium, and also we find Rhizopus. So all these agents we can do more studies, we can do more research to see how and what kind of strains of the agent that can be one agent as a biocontrol for this pest. Okay, so for this presentation, that what I want to inform that basic study is really important. So this is one of the basic study that to lead us to more understand and to more know what kind of things that we can do. But yeah, maybe many research already do like this, but we need to do more studies to enlighten the entomopathogenic. Thank you so much.
Okay, thank you, Amelia Subayang and the team. Round of applause for you. Congratulate for all the speakers for this afternoon. There are 8 of them, and they have already delivered all the research works, very, very impressive research works. And now is the time that I will allow you guys from the floor who might have any questions or anything that you would like to say or ask from the presenters. So anyone have any questions or need some more clarifications, please? Somebody is raising his hand, her hand? No one? Are you sure? I heard that the committee provide a door prize for those who ask questions. Am I right? No one, really? Okay, this is for those who wear the blue shirts. You are still young and energetic, and I hope that you will ask questions, guys. Otherwise, I will hand it over to the gentleman over there who is not wearing blue shirts, not so young. But okay, Bapak, you have the mic with you already? Okay. Please introduce yourself and identify to whom the question will go.
Okay, thank you. Please introduce myself. My name Rudy Thompson Utasoid. I come from Tungro Research Disease Station, Landrang. Okay, thank you. I want to ask to Mr. Jainuddin and team. At the presentation, Mr. Jainuddin and team, the combination of biological control agent with dipenoconazole significant suppress maize leaf blight compared to the combination of BCA, biological control agent, and other synthetic fungicides. Now, can I get additional explanation of the factor that affect it? How biological control agent with dipenoconazole significant other combined fungicide synthetic. Thank you.
Okay, thank you, Pak Rudy. So that question will go to the team of Ibu Nur Asyah Jainuddin. It's actually an Ibu, Pak Rudy, not Mr. It's a Mrs. So, Ibu Asyah, please, you may respond to the question.
Okay, thank you. I will try to answer. Did you mean how to the biological control apply to the plant, or can you verification?
Okay, Pak Rudy is actually asking for one kind of biocon. How this I don't know what the name. What you may repeat? Yeah, that one. How this will effect on the other on the
biological control?
Yes.
In this research, there is no negative impact if the BCA or biological control combined with the synthetic fungicide, and because from the laboratory test, the difenoconazole which have the highest inhibition for the bipolaris maize, so in these field trials, we can what I say difenoconazole by spraying the at the plant, and the later we can spray BCA again, like that.
So meaning that this agency is the best form in the study. He is the best to control the bipolaris maize.
Yes, BCA combined with difenoconazole.
Okay, combined with the difenoconazole. Did you get the answer, Pak Rudy?
Okay, thank you.
Are you satisfied, or do you want to run after another questions? Satisfied?
Okay, I want to. Can I get explanation that affected factor why combine BCA, biological control agent, with difenoconazole effective, significant, and the other combined. Why? Okay.
Why is it this combination is the
It's
best?
okay.
Yes, because from the percentage of disease severity at the field trials, difenoconazole combined BCA was found the lowest disease severity infection to the severity of meadows leaf blight. So we can recommend it that difenoconazole combined BCA have compatibility to control meadows leaf blight, like that.
Yeah. So probably this compatibility mechanism is something to dig a little bit deeper for the next research.
Yes.
So what kind of compatibility is it? Something to do with the particle size, for example, or I don't know. It gives us an opportunity to explore more on the research, Ibu Asia. We will wait.
Okay.
Probably on the third ICFST we will be hearing the progress of this research.
Okay.
Thank you, Pak Rudi.
Yeah, thank you.
Okay, Bu Asia, thank you. And then any other questions? Oh, okay, Ibu. Please. I'm happy if people asking questions.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Please introduce yourself first,
Good
Ibu.
afternoon, ladies and gentlemen. My name is Reni. I'm trainer at National Agriculture Training Center. We call in Indonesia Balai Besar Pelatihan Pertanian Batang Kaluku, located Kabupaten Goa District, Sungguh Minasa. I want to ask Prof. Yusminah Kak Emi. long time we must children together, ya. Yeah, I want to ask why use fruit like tomato and purple passion fruit and strawberry, not another vegetable or another fruit. Yeah, that's all.
Okay, thank you. So Ibu Reni is asking Ibu Hala, her her old friends, why she's doing study on the fruits, not vegetable, and why those fruits are chosen, not other fruits. Durian probably.
And anti-aging, ya?
For the antioxidant. Okay. So Ibu Hala, I will request you to respond to the questions of Ibu Reni. Your old. So this is kind of reunion for you. Okay, please.
Okay, thank you, Ibu Reni Amril. Thank you for the question. I'm very glad to hear and meet you again in this room. Yeah, Ibu Reni is my friend when we was a child. And then we go to the campus, yes, dari Fakultas Pertanian 80 dan 81.
Wow.
And then we meet here. Okay, thank you for the question. Why we don't use vegetables to make fruit mixed vegetables? This due to the habit of the people who always mix the fruit and to use juice or smoothies. Yes, jadi the habit to make a juice by fruit, mixing fruit, this make a big question for us. How about the antioxidant content of the mixed fruit? Is it lower or higher than we use single fruit? And it's very good news that besides we get the simplicity of making mixing fruit, we also get the higher antioxidant level or content of the mixing fruit. Ya, gitu Bu Reni. I think that's all. Thank you.
Thank you, Ibu Hala. I hope Ibu Reni already satisfied with the answer. If you're not satisfied, probably you may ask her to do some more research on other fruits. Oh, yeah, probably because of the fruits, yeah. Okay, thank you very much. Any other questions from the floor? Oh, okay, this is my teacher, so Pak Willy will ask questions. So I have to run over and hand him the mic. Sorry, Pak Willy. Ya, monggo silakan.
So thanks very much. Good afternoon, everyone. I'm Willy Suwarno from IPB University. I'd like to ask a question to Mas Tarmizi. Yeah. It's a very interesting research about anthocyanin in corn. Really appreciate it. And then my question is, why do you choose the exponential model for ASI? I mean, would it be another model that might be more appropriate for it? And then the second question would be, would it be possible to include another X variables to better explain the Y variables? I mean, but this result is really impressive to me because this model can explain even more than 50% of the variability of the grain yield, I think. Thanks very much.
Thank you, Pak Willy. So, Pak Willy, for your information, the presenter is actually Pak Yasin. Yeah, Pak Yasin Hage. He is the former researcher of Indonesian Cereal Research Institute. So I may hand. Oh, sorry. So okay. He is one of our breeder in ICERI, especially for special corn. Pak Yasin, yeah. You may respond to the question.
Thanks, Dr. Willy. So me and Tarmizi would be explained the experiment by Tarmizi. We select the exponential model ASI. This is the reference from the Dr. Peck from the CIMIT. So when training today, he gives instructions to me, you don't use simple regressions or multiple regressions, because this is the model which is the best for the exponential if you want to predict it of the yield by correlation with the anteceding interval. The first I tried to the simple regression, it's very simple. So of the one abscissa and one ordinate. Tapi he told to me don't use the simple regression or multiple regressions. You have to use the exponential models. So you transformation to the logarithm E 2.7138 and 5 before you make the prediction of the models. So the second question, we observed many variables, but for exponentials, I see the reference also came from CIMIT, not important the plant height, year height, and so on. You concentrate only the copse of the result, or all character of the copse you observed and make relation with the AC It's after explained the number of seed for one line, number of seed for one egg, the length or diameter of the ear and so on. Those seven parameters after explained in the national seminars. Not important the another variables if you make to the exponential model of the RC. So you concentrate on the ear, the result. Thanks.
Okay, thank you, Pak Yasin. Pak Willy, are you satisfied with the answer? Okay, thank you very much. Yeah, the antasis silking interval is something tricky, yeah, Pak Willy. Sometimes it doesn't really affect the yield. Sometimes it's totally ruin the yield if you have a long ASI interval. Yeah. Okay, thank you very much, Pak Willy and Pak Yasin. Any more? Any other? Okay, if there is no more questions, I would like to allow me on behalf of the committee to acknowledge everyone who already participated in this session, especially for the speakers, Ibu Hala, Ibu Ramlah Arif, and then Ibu Nur Asyah, Bapak Arif, Pak Yasin, and then who else? There are 8 of them, but I cannot I cannot memorize. So thank you very much for your sharing to us. Your research work is really impressive, and we are looking forward to hearing some more updates in the future, in the 3rd ICFST, for example, hopefully. And we are also would like to sorry or apologize for any inconvenience, some technical problems, some sort of discomfort on anything. So thank you very much in particular also for the IT team, for this cameraman here, thank you to take my photograph. So yeah, with this I will declare today agenda is over. You may go home or go back to your room to rest. Thank you very much, and we will see again tomorrow at 8 a.m. local time. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much. Oh yeah, one more thing. Amelia, are you still there? No? Okay. For those who need the certificate of oral presentation, only for the presenter, yeah, you may get the link from the committee. You get the link and then you click that link and input your name with the title, your institution, and then the title of your presentation. And then automatically the system will generate the certificate for you. So if there is any more clarification you need, you may contact the committee. Thank you very much. See you tomorrow, everyone. Good day.